Good afternoon and welcome everyone to this externally led patient-focused drug development meeting focused on genetic cardiomyopathies, including hypertrophic, dilated, arrhythmogenic, amyloidosis, Friedreich's ataxia, and Danon disease. My name is Lisa Salberg, and I'm the founder and CEO of the Hypertrophic Cardiomyopathy Association, which is a nonprofit patient advocacy organization representing those with thick heart muscle disorders. And my co-host is Genevieve Eccles, the Director of Family Support and Research at the Sudden Arrhythmia Death Syndromes Foundation. Today, we are honored to not only represent our own organizations, but also the Amyloidosis Research Consortium, the Friedreich's Ataxia Research Alliance, the Danon Foundation, and the Dilated Cardiomyopathy Foundation. Collectively, our missions are to advance the treatment of genetic cardiac disorders in our lifetime, and we do this by providing direct patient and caregiver support, high-quality education, supporting promising research, and raising awareness of the impact of genetic cardiac diseases. A special welcome to the many staff members of the U.S. Food and Drug Administration that are taking their time to be with us today. Thank you for giving us this opportunity to first hold the meeting. We're excited to have this time with you, and we hope that today you learn a lot from the patients and caregivers in the genetic cardiomyopathy communities. We want to give a special thank you to our sponsors, AskBio, Haya Therapeutics, Lexeo Therapeutics, Tenaya Therapeutics, and Rocket Pharmaceuticals. We are pleased to have in attendance representatives from the genetic heart muscle communities, professional organizations, pharmaceutical and biotech companies, federal agencies, and clinical and research centers from around the globe. Thank you for joining us today and for your interest in genetic cardiomyopathies. Most importantly, we want to welcome the members of our communities whose lives have been directly impacted by heart disease globally and cardiomyopathy specifically. Today's meeting is the result of many months of planning and people working together behind the scenes, some of us working together for the first time. We want to extend our deep gratitude to everyone who has had a hand in preparing for this meeting, and specifically to Professor Hugh Watkins and Dr. Christine Seidman, who've acted as medical advisors. We have had an extremely productive pre-meeting preparation, including the respondents are over 2,000 in our survey, and you're going to see some of those results today. The survey will remain open for 30 days after the meeting for additional comments. We are grateful to have this opportunity to ensure that patients and caregiver perspectives are considered in the drug development and regulatory process as we all navigate the new frontiers of genetic and RNA-based therapies for cardiac disorders. An estimated 2.3 million Americans have a form of genetic cardiomyopathy, impacting both the physical and emotional quality of life and running through generations within families. Treatments are currently limited for most cardiomyopathies to symptom relief and rhythm management, with targeted therapies being approved for obstructive hypertrophic cardiomyopathy with myosin inhibitors and for ATTR amyloidosis with RNA silencers and stabilizers. And for many cardiomyopathy patients, heart transplant is the only path to survival with its own complications and management burdens. Over the next few hours, we will discuss the burden of disease of a variety of cardiomyopathies, and our hopes are as a collective community for innovation that will bring targeted treatments that impact the root cause of our diseases, and dare I say, someday bring us a cure. And now we are going to hear from Dr. Erica Young from the FDA. Thank you. We're off to Dr. Young.
Thank you for that kind introduction. Good afternoon, everyone. I'm Erica Young, and I'm a clinical reviewer in the Center for Biologics Evaluation and Research, which is the section of the FDA that regulates cell and gene therapy products. Thank you to all the patients, caregivers, and advocates for being part of this meeting and sharing your experiences with us. I would also like to thank the HCMA and all the organizers and staff who were involved in planning this meeting. While FDA plays a critical role in facilitating medical product development through regulation and development advice to drug developers, we are just one part of a very complex process. We also have representation from clinicians, academic researchers, members of industry, and other stakeholders participating today. And I'm very excited to see a high level of interest from people involved in all aspects of drug development. FDA's patient-focused drug development initiative came about because FDA recognized that there was a need for more systematic ways of gathering patients' perspectives on their condition and different treatment options. In 2015, we started welcoming patient organizations to identify and organize their own patient-focused collaborations, and these externally led meetings provide an opportunity to hear directly from patients, patient advocates, and caregivers about the symptoms that matter most to them, the impact the disease has on patients' daily lives, and patients' experiences with currently available treatments. Nothing can replace someone's account of the day-to-day challenges that come with living with genetic cardiomyopathies, and meetings such as this strengthen our understanding of the disease and treatment burden, as well as the potential unmet needs. FDA shares the patient community's commitment to facilitate and expedite the development of safe and effective treatments for genetic cardiomyopathies, and we believe that patient input collected from these meetings adds immense value to drug development and our own clinical evaluation of new products. New therapies must meet the needs of people living with genetic cardiomyopathies, and patient input can help to identify areas of unmet need, including symptoms that are most burdensome to patients or impact a caregiver's life the most. Patient perspectives may also help developers as they identify or create tools used to measure the benefit of potential therapies. Hearing from patients and caregivers about what they hope to achieve in terms of treatments and future trials is a vital resource that will inform our approach to the evaluation of clinical research. Although FDA does not develop medical products or conduct clinical trials, it is our responsibility to ensure that products that are approved are both safe and effective for a specified population, and that the benefits of those products outweigh the risks in the indicated population. The input that we hear from patients is extremely valuable for us to understand how patients view the benefits and risks of therapies for genetic cardiomyopathies and how they may weigh them against the unmet needs and against uncertainties about the disease or about the products. Following this meeting, we look forward to sharing the summary report on the FDA website, which will serve as a reference from today's discussions and help inform our future drug development programs for genetic cardiomyopathies. We are all here today to hear the voice of the patient, and we are grateful to each of you for being here and sharing your personal stories, experiences, and perspectives. Thank you, and I'll send it back to the studio.
Thank you, Dr. Young. We look forward to working with you and the other members of the FDA to bring new treatments to our patients. Now we're going to hear from the director of the Cardiovascular Genetics Lab at Harvard, Dr. Christine Seidman, for an overview on cardiomyopathies. Dr. Seidman, off to you.
Well, good morning. I'm happy to be here with you today and to tell you about our understanding of genetic cardiomyopathies. I particularly want to welcome all the patients and caregivers, and we look forward to your comments and thoughts. What we're going to talk about today is the genetic basis for heart disease, but we'll start by how the normal heart works. Genes, as most of you know, encode RNA, which are the ways in which proteins are made in each and every cell. Those cells become organized in a very specific fashion in the heart so that they can perform with high resolution, and that allows our hearts to pump properly throughout life. These are an extraordinary, complicated series of processes that require a lot of different genes and different mechanisms to execute throughout our lives. We know, unfortunately, that these normal functions can be perturbed and the heart muscle can become dysfunctional. When that occurs, the normal structure of the heart is very often changed. And in general, when the heart muscle is perturbed, we call that a cardiomyopathy. These come in two different flavors. In one, there's hypertrophy, in which the walls of the heart become thickened, but the function is generally pretty good and often normal for much of adolescent and early adulthood. Alternatively, other disorders can cause the heart to dilate, in which the volume within the heart is expanded and the walls appear to be thinned, but most importantly, the function is diminished. As you can see at the bottom of these slides, the prevalence of these disorders are very, very high in the general population, with cardiac hypertrophy found in 1 in 500 individuals and cardiac dilatation in about 1 in 250 individuals. The causes of these kinds of changes are many, but today what we're going to focus on are genetic ones. And these are particularly important because they cause considerable symptoms. They can cause palpitations, which we know represent arrhythmias. They can cause sudden cardiac death, and also heart failure. Heart failure is an increasing economic burden across the US and obviously has considerable society and personal and familial impact as well. It's estimated in the United States that in 2030, we will expend more than $70 billion in caring for patients with heart failure. So, for many years, we felt it important to understand the causes of heart failure. And to that end, a lot of work has been invested in understanding what patients present with and what happens along the course of their disease. And what we now know is that patients can prevent in many different ways. They can have the symptoms I mentioned before: shortness of breath, palpitations, sometimes some chest pain, or actually fainting or an absolute cardiac arrest. But often these individuals can present because their doctor detects a heart murmur or hears some crackles in their lungs, or they're aware of swelling in their lower legs. These will lead to clinical assessments, often a detailed history about the patient as well as the patient's family, physical examination, an electrocardiogram, and imaging studies of the heart structure so that we can detect without radiation the size and structure of the heart muscle. When that is felt to be something that has been passed down from parent to child, or it occurs at a higher frequency than we would expect, we then suggest that this may have a genetic underpinning, and that may prompt additional testing. Sequencing, in which a series of genes known to cause dilated cardiomyopathy or hypertrophic cardiomyopathy are specifically sequenced. More generally, we're turning to an analysis of all protein sequences encoded in our DNA, which is called the exome. And that allows for every different potential cause of a genetic cardiomyopathy to be developed, not just ones that are listed from prior knowledge. And increasingly, we'll see the use of whole genome sequencing, which will not only look at protein coding sequences, but the sequences that regulate the expression and the amount of those proteins as well. Genetic testing can give rise to several different results. Sometimes we find that there is a cause that can be identified as pathogenic or likely pathogenic in a particular gene to cause a genetic cardiomyopathy. These are actionable diagnostic results, and they allow us to then proceed with certainty in terms of treatments and assessments of family members who may also be at risk. Alternatively, a variant can have unknown clinical significance. It's different from what we generally see, but we really don't have the ability to interpret of whether it's damaging or if it's tolerated or what we call benign. And the last result would be one that is negative. There really are no significant changes that are detected. When we look at these different opportunities in the context of patient care, then we see that there are many different results that can then be initiated. One would be that because of the genetic information, we now understand the symptoms and can now assess that those genetic abnormalities are giving rise to the shortness of breath, the palpitations, the heart disease, and like. Alternatively, we can recognize that they are changing things that are found by either an electrocardiogram or the cardiac imaging, and as well can give rise to subsequent events that we can follow over time. But equally important, the precise genetic etiology can alert us to looking for other additional symptoms and signs. We know that given the variety of different genetic causes of cardiomyopathy, that sometimes there are additional manifestations in the central nervous system, and that results in neurologic symptoms. ataxia or a spasmicity of our nerves and muscles to function normally, neuropathy, where there's pain and discomfort, and sometimes intellectual disability. We know that some of these that variants can also affect not only heart muscles, but skeletal muscles as well. And more broadly, they can affect the gastrointestinal tract, the kidneys, and our eyes, as well as a variety of other systems throughout the body. So, understanding the precise gene that is causing the common symptoms of cardiomyopathy and those that might be specific to a particular disease can help us not only to address the cardiac abnormality, but the associated manifestations. Although we know of now many genetic causes of cardiomyopathies and a variety of different manifestations that these can evoke, the medications that we use are pretty much all the same across the stages of disease. And these include beta blockers, calcium channel blockers, and sometimes antiarrhythmics to deal with palpitations and serious arrhythmias. As individuals progress, we may add ARBs or ACE inhibitors, diuretics, and when there are important risks for embolic events or clots, we use blood thinners. Antioxidants and mitochondrial medicines are sometimes added to the regimen to increase metabolic capabilities of the heart. And increasingly, we're using drugs that were initially developed for diabetes and obesity, SGLT2s, because of their benefit on the myocardium. When we advance to surgical procedures and interventions, we know that one of the most common is an AICD or an internal defibrillator to prevent lethal arrhythmias. Other arrhythmias we can treat by ablating them in the electrophysiology lab or by a surgical procedures, and with sometimes the need for more advanced interventions such as sympathetic denervation. When the tissue appears to be damaged in a way that obstructs function, we can remove it with a septal reduction approach for hypertrophic cardiomyopathy or repair a leaky valve, as sometimes occurs with dilated cardiomyopathy. More advanced therapies obviously would include the assist devices that are placed into the left ventricle or ultimately cardiac transplantation. Now, there are some specific and more highly targeted therapies, and these include myosin inhibitors that are largely used for hypertrophic cardiomyopathy and appear to have benefit in particular for genetic cardiomyopathies, but also for some without a known gene mutation because they have the same manifestations. For amyloidosis, there are two different ones. They're called TTR stabilizers and TTR silencers. And in addition to this regimen, as we'll talk about later on today, there are increasingly genetic therapies with enormous opportunities to impact the health of our patients. Most importantly, I want you to remind you that despite these interventions today, we now know that those patients with genetic cardiomyopathies have more adverse effects than those who have dilatation or hypertrophy, but without a genetic cause. Shown on the left here is a comparison of adverse events or the freedom from adverse events in people who do not have a mutation, and those are shown in red. They have fewer adverse events than people who do carry a sarcomere mutation, and they would be genotype positive. On the right is the dilated curve, in which a variety of different mutations are shown. And again, you can see that while they vary, all of these are significantly higher in terms of the risk of a cumulative adverse event in comparison to people with non-genetic dilated cardiomyopathy. So, with that, I'm going to stop, and we look forward to hearing from all of the amazing stories that our patients and caregivers have assembled to better understand these genetic heart diseases and genetic cardiomyopathies. Thank you.
Thank you, Dr. Seidman. We'll be hearing more from her later on future therapies. And for now, we are going to look at some of our survey results. And for that, we have with us today Joey Graham with the survey results. Joey, off to you.
Thank you, Lisa. I want to just say I'm happy to be here. I am a patient. I have HCM, and I'm happy to be giving the responses today of all of the folks that took the time to fill out our questionnaire. So let's first take a look. Who are we and where do we live? And you will notice by looking at this pie chart that the majority of the respondents who took the time to do the survey are from the United States. But we did get some light bulbs that started to register from other countries. We can see Canada on there. 2% of our respondents came from Canada. We had almost 5% from Europe. And look, we even have some other countries that came in, like Australia. So we ask, have you, your family, or the person you are completing the survey on behalf of had genetic testing? And if so, what were the results? Look at that green line. It's almost 50% said yes. We have had genetic testing and a pathogenic mutation variant was identified. The test was positive. Look at that line, 50%. So step down, we've got about 13% that said yes, we have had genetic testing and a variant of uncertain significance, which they abbreviate sometimes as VUS, it was identified. About 21% said yes, we have had genetic testing, but no variant mutation was identified and the test was negative. And you can see on the results there that we've got about 4% that weren't sure and about 12% with no testing. Now, let's look at this question. Please express your relationship to cardiomyopathy, genetic heart disease. The majority of folks who responded, about 87%, they are an individual living with genetic cardiomyopathy or have related heart muscle disorder. About 7%, a parent or legal guardian. About 5%, a person that has a family member or loved one with genetic cardiomyopathy or related heart muscle disorder. And you'll see there just a tick over 1% that said they do not have genetic cardiomyopathy. How old are you is the next question, or the affected individual. Let's look at these ages. As you might expect, the youngest, we've got under a percentage that are currently affected, but those numbers start to tick up. 6 to 18, you got 4%, about 5% from 19 to 30 years old, 31 to 40, about 8%, 12%, 41 to 50, 22%, see how these keep climbing? The older we get, 51 to 60, and look at that number, the pink line there, about 50% of the folks who responded to our survey are older than 60 years old. If you're looking at your screen here, you're going to see a lot of colors, a lot of numbers, and a lot of letters. And we're going to go back to our experts at the desk to say exactly what we're looking at here. A lot of things represented on the screen here. So Lisa, back to you.
Joey, thanks for those survey results. So as you can see, we have a lot of different genetic mutations listed here. And the fantastic news is out of the 2,000 respondents to our survey, we have every identifiable gene for cardiomyopathy represented here, from the most common being MYBPC3 to the most rare, probably JUP and DSC2. So we have the ultra rares and we have the more common genetic variants that are here today. And on that note, I'm going to pivot over to my co-host, Genevieve, and we're going to hear a little bit about why you're here and some of your connections to genetic cardiac disease. And we're going to talk a little bit about our program.
Thank you so much, Lisa. I'm very happy to be here today. My name is Genevieve Eccles. I'm the Director of Family Support and Research for the SADS Foundation. And I'm also living with an inherited cardiac condition myself and with a defibrillator. We are very excited to be here to be representing patients with genetic heart conditions. Thank you, Lisa.
Thank you. And we are now going to be hearing directly from the patients on their burden of disease. And first, we're going to hear from Bo. Off to you, Bo.
I'm Bo Nemelka, and I live in Sioux Falls, South Dakota, and I'm 38 years old. It was about 1966. My grandfather, Mark Nemelka, was playing baseball, and he died in center field, throwing a ball from center field to third base. At the time, they didn't know what it was. They documented it as a heart attack, but they didn't really know. My grandfather had 8 siblings. 5 of those 9 children died under the age of 50, all from sudden heart failure, and most of them playing sports. So we knew as a family through the 70s, the 80s, and early 90s that something was going on, but we didn't really have any solutions and no way to really know what was causing this sudden death. So in 2018, I chose to be tested, and I did test positive. And we have a mutation that goes back to the Desmond protein gene. So they did a cardiac MRI in 2021 and found that I had light scarring. And they attributed it, of course, to the Desmond protein genetic mutation. And so I decided with my wife that it'd be best to get an ICD implanted, because in my family, the biggest risk is that your heart suddenly stops, particularly playing sports. About a year later, I was up at a boys' church camp with my son and his friends, and I was playing flag football, and I started to see stars, and I was shocked for my first time. The next 3 months, I was a little more careful, but found myself playing tennis again about 4 months later, and I got shocked again playing tennis. The next morning, I wasn't feeling well. And so my wife drove me into the doctor finally. And on the drive to the doctor, I got shocked again in the car. And so it was just one of those times where, okay, it was getting a little more serious. So we had all 5 kids tested. We decided to do that. My wife and I believe that we would rather know and be preventative than not know and have the worst happen. So we had all 5 kids tested. We knew they had a 50-50 shot at getting it, and 4 of our 5 children tested positive. I was 10 years old when my dad had a heart transplant for the first time. I don't think any of us really remember much when we were 10, but you remember how you felt. And I remember feeling scared, and I remember feeling worried, and I remember feeling like, I could probably lose my dad. And that was a hard thing to go through, but it's nothing compared to when you are the dad. And when you now think about life differently, because you have, for me, I have these 5 little kids who I care so deeply about. Your body and your brain tell you to be active, to play with them, to throw them in the air, to wrestle them, to just be with them in an active physical way. And to want to do that, but not be able to, it's hard. And I would say that to me, the hardest part of the last, 5 years, having gone through 3 shocks and, everything that comes with that, I would go through that 1,000 times again if it meant my kids didn't have to go through it. And I would 100% take any chance that would be given to me to avoid my kids having to go through the same reactive interventions, because there's no proactive interventions available. And I have one son in particular who has special needs. He was already born with something that he's going to have to manage that the rest of us don't. And it was very emotional for me when we were receiving the genetic test results, and my oldest son said, "Dad, I'm okay if I get tested positive if it means that Eli isn't positive." And that really hit for me, that his brother would actually be willing to go through what he's seen his dad go through and his grandpa go through, so his brother didn't have to. And unfortunately, that son did test positive. But just that sheer act of sacrifice and willingness to go through a hard thing, I don't think my oldest son would have said that had he not seen how hard it was for me. And that's tough as a dad, because you want to be tough. You want to be that dad that they look to, like, my dad's invincible. But when they see dad in bed a lot, or they see dad a little bit down, or not acting like his normal active self who used to coach the baseball team, they know. they know something's wrong. So, yeah, if there was a future where I could have an option available to me for a therapy that provided me a better quality of life, great. But I'd do it more so that they could have that quality of life and that my children and my grandchildren could actually learn from what we're learning now. It's just remarkable to me that there aren't more interventions or ways that I can, as a parent, help my children think about this future and how to help it when we've known about this now for decades and the science should be more advanced than it is. So, as their dad, I feel like it's an imperative that gene therapy becomes part of the normal conversation in cardiomyopathy and other heart or cardiological conditions, because if not that, then we're all just playing the preventative game, which is highly expensive, highly disruptive to one's lifestyle. And although it saves lives, it doesn't necessarily position, I think, for the most healthy and most productive lifestyle, which is something I think the FDA and others could actually change.
My name's Gretchen Anderson. We live in Clifton, Virginia. My son is Sean Anderson. He would have been 35 last year. Sean was actually diagnosed on the later end of, and he was symptomatic on the later end of childhood onset for Friedreich's ataxia. He just kind of tootled along for a few years with not having any kind of heart issues. We found out at the age of 23 when we went in for his yearly echo that not only had his heart issue gotten worse, he was now in DCM. So Sean was obviously got tested every year with an echo. When we went to see him, pretty close to when we went to see him, Sean's ejection fraction had dropped down to like 25, which is bad. It was at that point which they decided that they were going to go ahead and do an ICD, because even though he had like no arrhythmias that they could tell, because the risk of having the surgery was less than the risk of having a cardiac event. So each year from 2020 until 2025, Sean's ejection fraction rose, which is really unusual in a patient that has Friedreich's, because once they hit a certain point, a lot of times they just kind of fall off a cliff, right? And his neurologist was like, yeah, this is not usual. In February of 2025, we all got the flu. We all got the type A flu, which is the bad one. But the following week, he started to have tachycardia. He ended up having atrial flutter. So that was Saturday. We went home, and early in the hours between Monday night and Tuesday morning, he started throwing up blood. So we raced him back to the ER, and we got there. And then all hell broke loose. We knew it was serious. I mean, we knew it was serious when he started throwing up blood. What we couldn't understand, and the doctors at the end couldn't understand, was how he went from Saturday being perfectly in normal limits to being way out of them on Tuesday morning. So pretty late, they were trying to wean him off the medications, and pretty late in the night, the nurse said, he's starting to come around and he's starting to move. And so I went over, they go, he's opening his eyes. And so I went over to him and I held his hands, and his eyes were a little bit open. And I was like, Sean, this is Mom. Look at me. Open your eyes, Sean. And he opened them for a little bit, and then he closed them. And I thought, yay, everything looked okay. And I thought, great, we're going to start doing this. And I was asleep in the room, and I got woken up really early in the morning by people running in the room, because his heart started to go crazy again. It was just tachycardia. He was in atrial flutter and AFib. And at that point, his heart gave up. His blood pressure was 80/80 or 70/70. There was nothing we could do. So we called everybody in that had come back, and we all said our goodbyes. And Saturday the 15th, we let him go. And the thing is, they can be okay for so long, but it just takes one bad something and they're gone. And that's the problem. They're so fragile. He loved several things in his life. He loved chess. He loved his birds, his parakeets that he shared with his grandma. Most of all, he loved fishing. As soon as he could hold a pole in his hand, Sean fished. He loved living back in Virginia. There are so many streams. There are so many ponds. There are, I feel like he walked longer than he would have because he was so determined to get to every point to fish. He would practice fishing with tying acorns to fishing line and fishing in the driveway for squirrels to see if the squirrels would take the acorns. So he beamed when he was fishing. The other thing I want to tell you about Sean is Sean was really, really good from the time he was young with kids that were younger than himself. I mean, that was just the kind of person he was. He just was like a Pied Piper for little kids. They just adored him, and he was so good with them. I think he would have been an excellent father. Sean really believed, and I did too, that gene therapy was going to be instrumental in helping him fix his heart. And hopefully before it's not too late, that's what we felt. But if someone is incredibly ill with cardiomyopathy, they should be able to try. And I'm here speaking to you because I do not want one more parent to have to live with what I have to live with. I don't want one more parent to lose a kid. When we started on our Friedrich's journey, I was looking for a cure for Sean. But very soon I realized it's not just about Sean. I'm here for all of them. We need to find a cure, and genetic therapy is probably our best chance of getting there.
Hi, I'm Giselle. I am a patient living with hATTR amyloidosis, and I have the gene variant V122I. I live in the Chicagoland area. I was born and raised in Chicago. I inherited this condition from my dad, and this is him, my twin, my fraternal twin, as my mom called us. I have grown up here, and I enjoy a vast of activities, skating, bowling, and dancing, and I love to travel. So living with this disease has slowed down my activities, but I am going to continue as long as I can, enjoying life and enjoying everything that I love to do. My dad and I were thick as thieves, and I walked in this house one day and he told me his feet were swollen. Took him to the hospital, found out he had hATTR amyloidosis. My dad's discovery of his condition, he always said, I just want to know what's wrong with my heart. At that time, when I looked it up, there was not much information. The only treatment was a transplant. And at him being almost 80 years old at the time, it was difficult. But it just made sense that all the symptoms that I've been having for over 10 years may have been what he was going through as well. So all these diagnoses became symptoms of hATTR amyloidosis. So my journey actually began with my dad getting diagnosed, me getting a genetic test. And then I did a PYP scan that discovered I did have amyloids in my heart. So from there is when we moved forward with starting the treatment process. My cardiologist is who is my amyloid specialist, and she is also a clinical trial doctor for amyloidosis. She gives me all the information about the treatments that are current, the ones that are on the horizon, and those that are coming in the future. So I'm very well informed about the condition, along with Google that we all use. But I started off with infusion of Onpattro, which was every 3 weeks, and it lasted 4 hours. So the same company came up with an injection that is every 3 months, and I'm in and out in about 15 minutes. I also was on an oral medication called Vendamax. It's no longer being covered by my insurance because they feel like I have enough treatment. But there are clinical trials on the horizon now to change that 3-month to a 6-month treatment, as well as treatments that will reverse the condition or stop the gene from producing the bad amyloids or the bad protein. So I'm looking forward to all of the research to continue and for the approval of these treatments coming in the future, and maybe even reversing it completely and reversing all these dreadful symptoms that I still have. Being an only child and not having children, I didn't have to worry about passing the disease along. But my dad's older sister had similar symptoms before she passed and never was diagnosed. I have 21 first cousins. The day that I officially was diagnosed, I sent brochures, text messages, emails to all my cousins so that they can get tested. I've had 3 that have been tested, 2 were positive, and I'm working on the other ones to get tested as well to find out if they have been dealing with the symptoms of hATTR amyloidosis. The United States thrives on diversity. In these clinical trials, we need to see diversity. We need to see the female population, the African American population, any of the diverse groups in this country needs to be included in all the FDA trials for this disease because it affects everyone's household around the world.
Hello, I'm Ashley Fisher. 5 years ago, I spoke to you as a patient living with hypertrophic cardiomyopathy, not knowing what was ahead for me. Today, I'm speaking as a heart transplant recipient and as a mother watching her 18-year-old daughter continue living with this disease. In 2020, I was managing symptoms and trying to live a normal life, at least what a normal life looks like for someone with HCM. After giving birth to my son, testing confirmed that my condition was progressing to the point that I needed to have a myectomy. I had class 3 heart failure with shortness of breath from walking up just 1 flight of stairs and constant fatigue. It was only after having a significantly preterm delivery, undergoing a myectomy and recovering from such a major surgery that I fully appreciated that my HCM was an aggressive type, which was confirmed by genetic testing. All the disease management I had done by staying hydrated, giving up alcohol, and taking medications religiously was never going to put me in control of HCM. I've learned that in spite of having an ICD and being fastidious about my lifestyle, I'm not guaranteed a symptom-free, healthy, or long life. But I, like any parent, want to be around as long as possible for my kids. Within a year, my condition progressed to advanced heart failure. And in 2022, I underwent a heart transplant on the same day that my daughter turned 14 years old. Transplant is not a cure, it's a trade-off. It means lifelong immunosuppression, ongoing risk, and a future that is still uncertain. While I was recovering, my daughter's journey was also continuing. In my case, and specific to our discussion, I need to be present for my 12-year-old daughter who gene tested positive for HCM and already shows some hypertrophy on echo. What I want for her is a life that allows her to be a runner if that's what she wants, or to keep skateboarding without feeling winded. When the time comes, I want her to have a healthy full-term pregnancy and delivery. If she takes medicine to manage her HCM, I don't want her to feel tired or sapped of energy every single day. I don't want her to fear that her medicine is not doing enough to improve her symptoms or to save her life. As she recently told me, "Mom, I don't mind if I have to take pills like you, but I want to be able to run around with my friends and not be out of breath." Thank you. We share the same genetic mutation, MYH7. She has already undergone open heart surgery, having had a myectomy last fall. She now lives with an ICD as well. Despite everything she has been through, we still cannot stop this disease at its source. What's hardest is not just what we've experienced, it's knowing that medicine today is still largely reactive. We wait for symptoms. We respond to the damage that this disease does. We intervene after the disease has already changed our hearts. That needs to change, and that is why I am here today. We need to connect great science with practical medicine to deliver treatments that truly change the trajectory and the quality of our lives. That means advancing genetic therapies and designing clinical trials that reflect the realities of patients and families living with this condition. My story did not end with a transplant, and my daughter's story is still being written. Like I said, she's 18 years old. One day she'll want a family of her own. So what will the rest of my daughter's life look like? And what will her children's lives look like? We need options that will change their stories. Thank you.
Some of those stories are inspiring and some of them are heartbreaking. And I think it's really unique that we brought you a follow-up almost 6 years later, and that's how fast HCM can change in a family and all the genetic cardiomyopathies can. I'm going to do something a little different for a PFDD, and I'm actually going to bring you a little bit of real anatomy. And that real anatomy is in the form of the HCMA's plastinization project. And I'm going to be showing you some anatomical hearts right now and talking a little bit about some of the patients that were impacted. This heart belonged to a woman in New York. Her name was Irene Lindenberger. Irene had no cavity left. She did not have a known mutation for HCM at the time of her passing in 2018. But if you can take a look in that heart, there is literally no ventricle left, and there's a lot of scarring and fibrosis within the heart. Then we have Victoria Teresa, who lives in Michigan. And this heart is very thick, as you can see. She was provided with a myectomy by a local physician, but it didn't really take a lot of the muscle out. And she was left to progress to transplant before, or approximately at the age of 30. She had no identifiable mutation either. Another heart that I'm going to bring to you is actually the daughter of the founder of the Cardiomyopathy Association in the United Kingdom, Amy Mann. Carolyn was her mom and my mentor. This was Amy's heart, and she transplanted. See how narrow her ventricle is and how thin that is and how hard the blood had to work to get from the bottom to the top of her heart. It's quite an unusual anatomy. Next, we have a double gene mutation heart. Her name is Hannah Wolf, and she's currently residing in Boston, but was down in the South for many years. This is a myosin-binding protein C3 and a troponin I mutation combination. She had a myectomy as a teenager and transplanted in her mid-20s, and today is practicing as a nurse and as a nurse navigator for the HCMA. So she moved into more of what would be like a dilated state of cardiomyopathy, and at the end of her heart, her ejection fraction was about a 25. The next heart that I'm going to show you is literally a world-class heart. It is a Danon patient. His name was Jeremy Acosta. Unfortunately, he did not make it to transplant and passed away in 2007. Please disregard the black. That is because it was in formaldehyde for many years. The sheer volume of this heart weighed 1,416 grams, and it was published as the world's largest heart at that time, Bill Roberts and Barry Marin. And you can see the defibrillator lead very clearly in the back of that heart. The next heart is a little unique because not only am I your host and the founder of the HCMA, I am also a patient with hypertrophic cardiomyopathy, and I had a transplant in 2017. And this is the thing that caused me all my trouble. So HCM is different. Every heart that I showed you could have been called HCM on an echocardiogram, with LAMP2 being the outlier, but they still call it HCM. And as you've seen by these anatomies, they're all very, very different. But we need to get to the basis. This is a myosin-binding protein C3 heart, and that is what runs in my family, and we would love to find a way to stop it from wreaking havoc on the rest of our family. So that is my testimonial as a patient with HCM, and you'll hear more at the end of the program about my family history. And now we are going to pivot to, well, actually, first I'm going to tell you that if you are going to call us, and I'm going to give you a phone number, but we're going to get those calls coming in a little while. It is 703-844-3231. Again, 1-703-844-3231, or there's a comment form that you can complete. If you're going to call us, please use your first name, city and state, or country. You don't have to provide any other identifying information. And please be assured that your comments, if we don't use them in the live version, will be in the Voice of the Patient report, which should be available October 2026. And we are going to include everything that you have to say. It's very important that we hear from the widest variety of patients possible. So you can start dialing and start getting ready to call. But first, Genevieve and I are going to look at some demographics, and then we're going to send it over to Joey for some more survey results. And if we could have that slide up. There we go. So we know, we talked a little bit earlier about how old the people were that were watching, but we asked them the question, when did you have your first symptom? And for many of us, we don't recognize the first symptom until way later in life, right? So here we have people at their memory telling them between 0 and 5 they had symptoms, or between 6 and 18, and you can see that this is pretty consistent. Every age is about 18 to 15% of people are identifying that they had symptoms that early. Now, Genevieve, let's look at the number. When did they get diagnosed? What do you think about that?
I think that really shows that there's so many opportunities to identify patients earlier. And one of the ways that we can do that is through genetic testing and making sure that we have that cascade screening, that family members are tested and they are identified early.
Absolutely. Now we're going to hear some more survey results from Joey. Off to you.
Thank you, Lisa. Which of the following heart-related health effects do you, affected person for the survey, experience as a person with heart muscle disorder? Let's just take a look at some of the top results we got. And the most popular response at 70+% was shortness of breath, closely followed by fatigue. We had about 60% of folks that reported arrhythmias and heart palpitations, and then about 51% coming in with lightheadedness, and we see then about 50% with a heart murmur. We ask, do you have a non-cardiac impact of the genetic disorder on other organ systems? And look at this chart. It's astonishing. About 50% of respondents said no. None of the above have an impact. We had about 30% say muscle weakness and fatigue. We had about 15% say gastrointestinal issues. And then you can see some of the other results and symptoms that they have. Let's look now at the top 3 most burdensome cardiac concerns that affect you. Over 50% had shortness of breath. 41%, just a tick over 40, came in at arrhythmias and palpitations. Look at this, about a third say exercise intolerance. And then I want to highlight this one too. Nearly 30%, almost a third, said they had concern about the development of cardiac symptoms, and that plays into the mental effects of this. And we'll end this round with looking at some of the top activities that are the most important and people are not able to participate in. And this, to me, as a patient, didn't surprise me, but it's good to see that it matches up. About 64% say just exercising is something they would love to do. Nearly 40% said participation in sports. 33, a third, said walking. And let's look at one more. 27%, Lisa, said social interaction and participation, one of the top things they're focused on that would improve their life. Back to you.
Thank you, Joey. I find it interesting that the bar is pretty low. Most of our patients just want to be able to walk and breathe. And it sounds like an easy thing, but for some of us, it's very hard. We're going to move to our Zoom panel now. We're going to hear some comments from actual patients living with cardiomyopathy. Genevieve, what are we going to be asking them about today?
So we have several questions, and we would love to as well as the panel that will be asking. We would love to have you think about these questions and call in and provide us with your comments and your experience. We'll be talking about all the symptoms and health effects of genetic cardiomyopathy, which symptoms have the most significant impact on you or your loved one's life. How does genetic cardiomyopathy affect you or your loved one on your best and worst days? How have you or your loved one's symptoms changed over time? How has the ability to cope with the symptoms changed over time? Are there specific activities that are important to you or your loved one that you or they cannot do at all or as fully because of genetic cardiomyopathy? And what do you fear the most as you or your loved one gets older? What worries you most about you or your loved one's condition? So now
Yeah, let's go to the panel and get them on up here. Hello, everybody. Thank you so much for joining us today. And thank you for being willing to talk about your personal experiences with genetic cardiomyopathies. So you want to start our panel off?
Absolutely. Thank you so much all for joining. So, Christy, of all the symptoms and health effects of genetic cardiomyopathy, which symptoms have had the most significant impact on you or your loved one's life?
Wow, Genevieve, there's so many, but let me just first start and say hello. I have arrhythmogenic cardiomyopathy. I have the desmoplakin mutation. I also have dilated cardiomyopathy in both my ventricles. So from a symptoms perspective, I'm run the gamut on what possibly could be there. I would say the most significant symptoms for me are palpitations and fear from what that means. I have had 4 major ventricular tachycardia episodes. And when I have palpitations, I have a lot of fear that I might go into sustained ventricular tachycardia. So the fear around that almost every day when I start the PVCs and PACs is still there, even though I'm 6 years post my original diagnosis. The other symptom for me that's really bothersome is I'm dizzy and lightheaded almost all of the time. And we can't really figure out why. I've been through vestibular therapy. I've been through all kinds of medicine changes to try to get it. But feeling like when I'm walking, I'm a drunken sailor, feeling very lightheaded and slow, exercise intolerance, which I saw just on the survey. So that's the other symptom that's extremely bothersome in my normal life. And then, of course, the effects on my life because of it, what I can't do, which I think we'll probably talk about in a little while. Thank you.
Thank you so much. Gwen, can we hear from you about some of the experiences you have faced throughout your life with dealing with hypertrophic cardiomyopathy?
Hi, Lisa. Absolutely. So I'm Gwen Mays. I have hypertrophic cardiomyopathy, obstructive, coming to you from the Eastern Shore of Maryland. And I just want to thank the FDA and all the listeners for their interest in this topic. It's really a privilege, but almost overwhelming for me to think about the segment that we're talking about today, because when I was first diagnosed, having had symptoms at birth, now almost 50 years ago, the first thing a cardiologist said to me is, "You will have a shortened life." And as a young woman without any Google, without any AI, I was devastated. There was really no understanding of this condition at the time, and it utterly shaped my life, the uncertainty of it. So I won't go into all the symptoms over the last 50 years, but I will say one that has remained consistent is a little what Christy hit upon, and that's the emotional toll or the mental health that comes along with symptoms. Symptoms will change. I get short of breath. I have fatigue. I get foggy headedness. I get not so much dizzy as I just feel fragile. I get hypervigilant. Everything that happens to me, whether it's a rash on my back or a sleepless night, I blame on my HCM or a medication that I'm taking. So I become sort of my own, not only advocate, but my own worst enemy when it comes to overextending or overanalyzing, being hypervigilant. But throughout all of those years, the uncertainty, the fear, the feeling isolated, also living with an invisible condition that is very difficult for those around you to understand when you want to take an elevator and everybody else is running up the steps, or you need to park in a handicapped space, even though you're not walking with a cane or using a wheelchair. The sort of emotional toll is something that has stayed and remained persistent.
For that, I think we might jump over to the phone in a second. We have a call coming in from Ross from Iowa. If we can get him on the line. Ross, are you with us? Not quite yet.
His son was not diagnosed.
Ross, are you with
He
us?
died, and he was never diagnosed.
We'll get back to Ross in just a second. Let me pivot over to Giselle for a second. Giselle, we heard from you earlier in the video portion, and you spoke about your father being diagnosed with ATTR amyloidosis and how that impacted your relationship with him. Can you talk a little bit about what his burdens of disease, his key symptoms were, and how you look at those symptoms and worry that that might be you too, or that is you at this time? Giselle?
Thank you. Of course, you touched on the most emotional part of my condition is my dad. I'm being an only child. We were very close from birth on. And as I tell people, mom said that I was daddy's baby and mommy's maybe because we were fraternal twins, so to speak. But my father was a humble person, never complained. So I walked in the house one day and he said, my feet are swollen. So pulled up his pants leg and looked like he was standing on tree trunks. That's how big his ankles were. So of course I rushed him to the doctor. They decided he had amyloidosis, but a couple of years of testing, they never determined what type of amyloidosis. Emergency room visit, they sent him to another facility. That doctor saw the symptoms and knew immediately that he was suffering with hATTR amyloidosis. And of course, the H stands for hereditary. So me being his only seed, of course, I tested positive for it. But his cardiologist called me personally after he passed in 2016, asked me to come in and get tested. But the key here is that when they finally got to him, his heart was only functioning at 20%. So the years of not being diagnosed, the condition lingering, and so on and so forth, early diagnosis is detrimental. So that's why his cardiologist called me immediately, asked me to come in and get tested. So I do have amyloids. They found them in my heart. They have not damaged my heart, but I have so many residual effects because hATTR is polyneuropathy and cardiomyopathy. And I suffer from vertigo, shortness of breath. There are times I can run up my stairs like an athlete, and sometimes I get to the top and I'm completely out of breath. One of the things I love to do is bowling. And I've been doing it since high school. Now, when I bend over to release my ball, I am out of breath and I am dizzy. So I have trigger finger, bilateral carpal tunnel, and that has slowed me from doing the sports that I've enjoyed for many years.
Thank you for sharing, Giselle. We really appreciate it. Are we ready on the phones with Ross now? Send him on in. Ross, are you with us?
Yep.
Hi.
Thanks for having me.
Thanks for calling in. So I understand you have a non-obstructed HCM?
Yes, I have non-obstructed HCM, and I had a long delay to diagnosis. But my topic, or the thing that I wanted to share, is about genetic testing. I'm MYH7, so I look forward to future treatments for that specific gene, because I don't believe there's anything currently in the market for that. And my mom and my son both have the same variant, and we are in Iowa. We are 3 of 503, so approximately. So we're really looking for answers. My journey actually led to AFib as well, which is really I struggle with more than my HCM. So I look forward for some sort of solution to AFib, because both non-obstructed HCM and AFib don't have really good answers right now, and I just hope that we can have a solution. And I know the PFDD can help the FDA, and I look forward to the FDA researching it more. So thanks for the opportunity.
Thanks for calling, Ross. We really appreciate your input. I'd like to pivot to Gretchen for a moment as a caregiver. We heard your story earlier. Thank you for introducing us to your son. Can you tell us a little bit about the caregiver experience with a cardiomyopathy? Your child had Friedreich's ataxia, which impaired his mobility as well as his heart. Can you share with the community what that felt like to be a caregiver?
So my story is a little bit different in that Friedreich's ataxia, he had fatigue, but Friedreich's ataxia comes with fatigue for most people. So it's hard to separate out how much of the fatigue is from the Friedreich's itself and how much is from his heart. Also, by the time his heart dilated, he was mostly using a wheelchair, so that kind of hides some of the symptoms. My biggest fear as a caregiver was, because he was so fatigued all the time, was that I would come in the morning to get him up and he would have passed. I have a couple of friends whose kids did. They had a sudden heart event and they came in and their kids were non-responsive. And it was really a blessing when we got the ICD. Maybe not for him, maybe he didn't like it as much, but it was for me, because I knew if something happened in the middle of the night that it would activate. And that was a real blessing for me, because I didn't worry so much anymore. I mean, I still worried, but like not about that particular thing.
Thank you for sharing that, Gretchen. We have another member of our community who also wrote in sharing this about her son. She said, I had no knowledge of my son having HCM, and he went into SCA and almost died from it, and now suffers a multitude of health problems. So I think that is such a huge concern for our parents in our group and our caregivers, is just that unpredictability of the condition and not knowing if your kids or if you are safe or not.
Last but certainly not least, Greg, you have a dilated cardiomyopathy story. Can you talk to us about some of the symptoms that you were experiencing and where your journey led you?
Yeah, thank you, Lisa, Genevieve, and all the folks at FDA and those that are participating. So I have dilated cardiomyopathy. My story is I was diagnosed with DCM, or dilated cardiomyopathy, in 2014. And being misdiagnosed, undiagnosed for 40 years, probably before that, and went through a lot of episodes for 7 years, and then was heart transplanted almost 5 years ago. And that's sort of my story. I'm in Columbus, Ohio. Started the Genetic Cardiomyopathy Awareness Consortium 3 years ago to address the lack of understanding of genetics and cardiomyopathy and the severe lack of genetic testing. Surveys show, peer-reviewed articles show, studies that despite guidelines from the American Heart Association, American College of Cardiology, and the Heart Failure Society of America, and suggesting that if you're diagnosed with idiopathic or non-ischemic cardiomyopathy, you should be genetically tested. Only 1.1% actually are, which is pretty bad. That is not taking place. I think it's something the FDA should know, and hopefully we can address. DCM is 50+% of all heart transplants. Once you're diagnosed with dilated cardiomyopathy, it's 50% survival rate for 5 years, which is pretty negative. I went through a lot of symptoms, experienced kind of everything, the gamut, but I'm happy to be here transplanted, and very lucky that I did not die. So
we're very glad that you didn't die either. So, and welcome to the heart transplant team, right? We are going to be pivoting over to the phones. We're going to go hear from Gordon from New Mexico. Gordon has an interesting scenario here where he is dealing with heart failure himself from a genetic cardiomyopathy, and he's also a caregiver to his wife who is dealing with other medical challenges. So Gordon, can you join us and tell us a little bit about your experience as being a patient and a caregiver simultaneously?
Sure. I'll just begin by saying that over the last couple of years, I've had increasing symptoms of heart failure, of edema, shortness of breath, and fatigue. This is following, I had a myectomy 19 years ago, and until a year and a half ago, those symptoms that I mentioned were actually pretty rare. They have several known causes. Two of them have a surgical treatment. One is I got leaky both aortic and mitral valves, and the other is I have badly positioned papillary muscles. And the difficulty is that none of them are bad enough to justify surgery by themselves. But between that and the stiffness of my left ventricle, I'm having more and more heart failure. And that means that there are days when it becomes difficult to do the things I need to do. There are times when it's difficult to carry out household tasks, whether that's cooking and cleaning or helping my wife just do various things, driving her to various events and things like that. I'll add that an additional difficulty is that the principal treatment for the heart failure symptoms is diuretic, which means that during much of the day I have to plan how far, I have to be keenly aware of where the next available bathroom is and plan how long it's going to take me to be near there. And so I'm less able to do those things than I used to be, and less able to exercise than in the past, and I'm concerned both that exercising may actually lead to worse heart problems, the lack of exercise, and the bigger concern is that it's going to become more and more difficult to care for my wife.
Gordon, could you talk a little bit more about the situation with your wife, if you're comfortable? What kind of challenges are you having there that make it maybe physically demanding for you?
Right. Well, okay. So my wife has Alzheimer's disease. She's at the, they categorize it as mild to moderate. She's at the later end of the moderate. She's got significant limitations in many things, and so the more than anything, I all of the household tasks, from cooking and cleaning to paying bills, are on me, and that means that cleaning, which everyone knows can be physically demanding, is physically challenging. My wife needs assistance with some physical things. And she's able to walk, but she walks poorly. And all of these things just contribute to just make it harder for me to be helpful to her, as well as make it harder for me to do anything else.
Gordon, thank you for sharing with us today. We really appreciate you calling in. And for anybody else who wants to call in, just a reminder, the phone number is 703-844-3231. Any more comments we want to hear from?
I would love to read this one from a parent who is writing in. They're saying, living with a genetic heart disease is difficult, but the most painful part is watching your own child go through it. My son is 14 years old, and beyond the physical symptoms, he lives with deep anxiety, fear about how the disease may progress, worry about his future, and especially the possibility of needing a heart transplant one day. At his age, he should be thinking about school, friends, and projects, not about life-changing medical decisions. This emotional burden weighs heavily on him and on our family. The symptoms, even when they come and go, create constant vigilance. Every chest discomfort, every moment of breathlessness, every dizzy spell triggers immediate concern. He struggles to project himself into the future or to feel like other kids, and this affects his confidence, his activities, and his daily well-being. Our greatest challenges are therefore both medical and psychological. Uncertainty, fear of deterioration, and the difficulty of reassuring an adolescent who understands all too well what is at stake.
Wow, that's a lot to take. I know I was diagnosed myself when I was 12 years old, and I was told by a doctor, believe this, that you can die suddenly and nothing can help you. CPR won't save you. It was 1980. There was no defibrillators available. So being a teenager diagnosed and being told your future is grim certainly makes you believe the physicians that are telling you that. And it is thankfully a new day, and we have many more therapies available. But the burden of disease to a teenager today is the same as it was years ago. You don't know what your future is going to be. And boy, wouldn't it be nice if we had a targeted therapy that can correct the heart muscle and have it work more normally, and we don't have to worry so much. So, Christy, you have a child, and you're dealing with worrying about your child. Do you want to talk a little bit about what it is to parent a child with HCM or with a cardiomyopathy?
Yes, absolutely. Thank you. So it was actually one of these things that I think a lot about the fact that we know, right? She was actually diagnosed because I was diagnosed with desmoplakin cardiomyopathy. She was 17 years old, and she had already quite a bit of damage to her heart at 17. The doctor did decide to wait a couple of years before implanting a defibrillator, and then did that. But being a parent, and so many of us are that are on this panel and that are in our audience, I know, it's a whole new level of grief and a whole new level of trying to figure out what are the very best things I'm going to be able to do for my child. And my child is a type A. She was going to run, she was going to do a triathlon her first year in college. And for arrhythmogenic cardiomyopathy, exercise is bad. You're supposed to move, but not do endurance exercise. You should look at the chart of exercise. It's actually kind of humorous when you look at bowling and archery on the list of okays. But it's one of these things that she destroyed her life, right, in the moment. And she has found ways to adjust, but parenting her through not only the physical surgeries of the ICD implantation, but also, and being there in the hospital when she had her first non-sustained VT over 3 days, that was hard physically, but the emotional challenges of having to work with a child on what their future looks like and how they're going to be able to obtain their dreams or change their dreams is huge. So for my daughter, she is still going to medical school. She's a third-year medical student right now, and I hope she sets the world on fire with her passion. But she cannot do the long-distance endurance exercise. She's had to change the way she looks at the world in terms of safety. Where is it safe to travel? What is it safe to do? And that's really hard when you're a young person.
That is challenging, but maybe she's going to be the one that brings us to a cure someday. You never know.
I hope so. Cardiology's on the short list.
That's a really good idea. We have another caller coming in. Sean from New Hampshire. We'd love to have you on the line with us. Are we ready with Sean?
Hi, how are you? Great to be with you.
Thank you for joining us, Sean.
So I'm a hereditary amyloidosis patient of a mixed phenotype, both cardiomyopathy and polyneuropathy. And what I'd like to discuss is the symptoms that are impacting me the most, or the ones that concern me the most. I do experience I'm kind of easily fatigued, not so much winded, but anytime I exert myself, I get tired and have to stop, within a minute or two. And the other thing that I have difficulty with is climbing upstairs, and I get fatigued by doing that too. And the concern I have is I don't necessarily know if it's cardio related or due to my neuropathy because I've got muscle wasting. So it's kind of it gives me a little bit of anxiety, but because I certainly am anxious that perhaps my cardiac condition is such that it could, trigger a worse type cardiac event for me. My best and worst days are on some days I'm full of energy and can get around and quite readily and do what I want. On other days, I'm kind of fatigued for the whole day. And again, with the mixed phenotype, I'm not sure if it's due to the cardio or not. So once again, some anxiety from that.
So, Sean, thank you for sharing. And if you could help our listeners understand a little bit more about being able to discern whether your symptom is from your cardiac condition or is it another external. This is a challenge we all have with cardiomyopathies. I think somebody already brought this up a little bit. Gwen, am I having a bad day because of my heart, or am I just having a bad day? And when you have something like amyloid and you've got all the muscle fatigue as well, it's really hard to be very clear about this. And I'm bringing this up very specifically for our friends at the FDA. When creating endpoints for clinical trials, we don't always know, and the clinicians don't always know, where our symptoms originate from. So Sean, hearing from you might help our friends at the FDA better understand how to maybe separate out some of these things. And maybe sometimes we don't have to. We can just talk about the general symptom and then let science figure it out over time as to what the underlying mechanism for that symptom was. Any comments on that, Sean? Oh, we lost Sean. Sorry, Sean. Thank you for the comment. But I'll take that question back to the rest of you. Giselle, if you'd like to comment in on that, I'd love to hear it.
Since I share the disease with him with the amyloidosis, he's absolutely correct. You don't know which symptom is going to hit you when. And it's a combination of the cardiomyopathy and the polyneuropathy. There are nights where I feel like I have sharp pains running through my right foot only. And it wakes me up at night. The bilateral carpal tunnel, bilateral bicep tendon tears, those are all part of polyneuropathy. But when I hit those stairs, of course, that's cardiomyopathy because I'm out of breath. I have palpitations at night. So his anxiety comes from not knowing what symptom is going to hit you when, because it's sporadic. And no amyloid patient has the same symptoms, and they don't hit you the same time, same place. I worked out regularly in the gym, and I couldn't figure out why I couldn't keep up with my friends anymore. And once I got diagnosed with this disease, it started to make sense. I was having issues with amyloidosis, cardiomyopathy. The heart's not functioning the way it is or supposed to. That ejection fracture is not where it's supposed to be. So until Dad got diagnosed, I had no clue. I went over 10 years trying to figure it out.
Thank you for sharing that. Here we have some comments coming in.
We do. We have one community member that talks about sitting on the sidelines of life. They say, my greatest challenges having to sit on the sidelines of life with the disease and not having a strategy to mitigate my symptoms or a cure. I struggle to get through each day. I exercise at the expense of my normal daily activities. I'm always tired. I'm not me. I'm thankful for my calcium blocker to prevent life-threatening events, but it's far from what I consider treatment.
I think all of us with cardiomyopathies can concur that sometimes the treatments just aren't enough. I want to pivot for a moment, and I'm going to ask Gwen to comment because I know Gwen focuses a bit on coping with the diagnosis. And there are a lot of different coping mechanisms that we utilize. It would be nice to not have to find all those coping mechanisms. But Gwen, can you tell us a little bit about not only your experience, but I know you run a discussion group for patients with cardiomyopathies and you focus on coping with it. And talking it out. So Gwen, let's talk it out.
Thanks, Lisa. yes, having a lifetime of a chronic illness, living alone, advised not to have children and never married. The responsibility really of keeping my wits about me and getting to the ER really has always landed on me. There's been a lot of resentment about that, a lot of fragility. Why me? A lot of shame when I eat pizza or have an extra glass of wine. I balance out living the life that I, Gwen, would like to live. It's pretty rambunctious versus what Gwen with HCM can live. And oftentimes they're at odds with each other. So I think the first thing with coping that I've had to accept over the years is that regardless of the length of time that I live, I want to live fully. I want to live to be a person that I enjoy, that I love, that I feel contributes to this world. And if that means that I push the envelope a little bit too much and spend the next few days in bed, that's exactly what I do. If it means that I have to get a caregiver or a friend to fly with me to a city because I'm too nervous to be by myself, that's what I do. If it means that I need to go on antidepressants, anti-anxiety medication, meditate, pray on my knees every night, that's what I do. So my responsibility, not only the physical challenges, but also making sure that I have the emotional stamina to deal with this condition is something that I take very seriously. I didn't always, I will honestly admit, but as I've aged, I realize it's equally important to me because it's enabled me to be a very good advocate for myself and for others. It's enabled me to comfort myself. It's helped me teach other patients. And so I really find that there isn't a pill for every person. As we hear, every patient's different. But I really just cannot say enough about the need and the hope that we as a community recognize this is a lifelong condition. This is a lifelong impact. And as Christy said, with her daughter, you have to rethink what's important. You have to reevaluate your priorities. You have to restructure your life. You have to refine yourself and still see that purpose that you have. And those are some of my thoughts about how living with this condition my whole life has had an impact on me.
Gwen, thank you. We have another caller. It happens to be one of my favorite names, but it's not my Becca, it's a Becca. We have Becca from Michigan who wants to talk to us about her 2 children, and we will be hearing from her later in the next segment of video, but I'd like her to be able to speak to you directly. Becca, are you with us?
Hi, thank you. Yes, my name is Becca, and I'm calling on behalf of my 2 children who are both diagnosed with Friedreich's ataxia. And like Gretchen, Gretchen's son. My daughter is 9 and my son is 5. My daughter was diagnosed at age 5, right on the edge of her birthday, and my son was diagnosed at age 2. And one of our first symptoms that led us to diagnostic testing was that she already at age 5 had hypertrophic cardiomyopathy, and as Gretchen had already shared, one of the most terrifying things is there's not a lot of information, and that there can be some major unpredicted cardiac events. So one of the most challenging things for our family is that we know that this disease is very hard on these kids, and especially when they're diagnosed early. So not knowing a lot of information, knowing that my daughter's heart had already started showing progressive signs at age 5, we are very aware of all the things that could potentially happen. And then hearing stories about, like Gretchen's, how her son was fine one day, and then just a few short days later, he had already passed, is terrifying. And so when our daughter, who, is running and playing, not running, she's walking quickly and playing, because of Friedreich's ataxia has already stolen her ability to run. But when she stops and she holds her heart, we are immediately in panic mode. We get out our heart monitors, and we're already preparing for some big issues. So there's a lot of stress and worry that comes with that on the daily basis. And then also, it's very hard to watch our kids just know that they have to be careful of their hearts. And when we go to the fair and they see signs that they can't ride a roller coaster if they have any heart complications, it's so sad to see a 5-year-old and a 9-year-old say, oh, I can't go on that ride because I have a heart condition. And we have to, go through all of those processes and just talk with them about what that is. And another fear of ours and theirs is that the hypertrophic cardiomyopathy will continue to progress and take away any other quality of life that hasn't already been stolen from them from this disease.
Becca, thank you so much. And we are going to hear more and see your beautiful children a little bit later in the program. I do want to take a moment here because we're using a lot of terminology in describing our cardiomyopathies. And you'll hear ACM and DCM and HCM. And sometimes we have crossovers. So you can have the same mutations causing both dilated and hypertrophic cardiomyopathy. You can see something that looks like hypertrophic cardiomyopathy and Friedreich's ataxia that then turns into a dilated cardiomyopathy. I showed you a heart earlier of a woman who had a very thick heart, had a myectomy as a child, and then went into an end stage where she dropped her ejection fraction to about 20%. So the reason why this PFDD is being focused on genetic cardiomyopathies as a whole community is because we cross over. And what will help one is highly likely to help another, if not an actual practical application of that particular agent, but in the concepts. And I think the ones that we've heard from already today give us a really clear indication that we are one big community, although we have segmented portions. Do we have any other comments we need to hear?
We do, and I think that this one really brings out the age range. We've heard from Becca and her children and Gretchen. We have a comment that came in. It says, before becoming symptomatic from HCM at age 59, I was very active. I worked outside all year for 40 years. We went mountain biking and hiking with the grandchildren. I restored old homes as an occupation. I was a regular to the outdoors and hardly did anything indoors. That has been completely turned upside down. I can go outside between the temps of 50 to 75, hard to breathe otherwise, develop major depression. I can only do 30 seconds of a 6-minute treadmill cardiac stress test. So these conditions affect all age ranges in our community, and they not only impact the physical, but the mental and emotional health of everyone involved, not only patients, but caregivers also.
Thank you. Greg, I'm going to come back over to you, and I'm going to hit you with a hard one, as you are also a nonprofit leader and you work with the community of dilated cardiomyopathy as well as your own family. I'd like to think a little bit more personally for a moment. What are your biggest fears for your family members? And what are you going to do to, let's see if we can change the future, but what worries you the most?
Great question, Lisa. Thanks. Yeah, I mean, it's time, right? So there are 3 genes, cardiomyopathy genes running in our family, lamin A, 2 lamin A genes, and a TPM1, which tends to manifest as hypertrophic cardiomyopathy. And so, especially with the lamin A, I've seen a lot of the major academic research on this say that, if you have the lamin A gene, there's a 90-some% chance that you will get the disease, right? When and how severe is up for grabs. It's pretty severe. I think ESC listed as the worst gene to get. So with my 3 kids having a lamin A gene, 8 grandkids not been tested yet, it's just a matter of time. And they will get sick. How soon, how severe, will they die? Will they need to get a transplant? But right now, that's the course for a lot of genetic cardiomyopathy, especially for lamin A. So the fear is, it's not even fear. I mean, they will get sick. The reason I'm doing what I'm doing is because speed to therapy development is so key. because there are, I think, estimated 1.5 million people out in the United States with genetic cardiomyopathy, and time, it ticks for my family. And so, but the fear is having them find their first symptom. They're being treated, right? And my hope and what I pray for and what I hope for is that all that we're doing, Lisa and your group and Genevieve and Christy and our group, is that we can speed the development of a cure, or there'll be multiple cures, and it'll come in waves, but time is of the essence, very much so.
I think you bring up a very good point about it. It's likely going to happen. We know penetrance of these genetic mutations is high, and when the disease shows up is variable. And this is going to be a challenge to our friends over at the FDA when they're looking at applications that are coming in. And we're talking about when to dose and who to dose and what families. This is complicated. And I'm not suggesting that we have the answer to provide for the FDA today and who, when, and how. But we know that our families, if we know the genetic mutation is there and we have history to lean on, in my case, my history can go back 130 years. And far too many people leave too soon. When is the right time to act? When is it too late? And when is it too early? Finding that sweet spot, we're going to need some time to work together to find that. And what we ask for is the grace to be able to have those conversations and decide, maybe not globally in this case, but patient by patient, family by family, when is the proper time to dose with new and novel therapies. So I really appreciate those comments. I want to pivot back to Gretchen for just a moment. Your son was lost, but his cardiomyopathy was found a little bit earlier than well, maybe a little bit later than some and earlier than others. I guess timing is timing. If you could have picked a time in retrospect of when you would have given him a cardiac medication that could have reversed, or a therapy, gene therapy, RNA therapy, would you have waited till he showed symptoms, or would you have wanted him to get it before there was anything visible? But what do you think would have been the right timing?
So Sean was on the late onset of childhood onset of Friedreich's. It's typically between 5 and 15, and he was 15 when he started showing symptoms. But by the time, a couple of years after he was diagnosed, so he was 17 going on 18, he was in the idebenone trial, and he wasn't having, he'd get chest pains, but we go in and it would be nothing. But when we were in the idebenone trial, one of the endpoints was heart wall thickness. So they were doing echoes on them, and he already had hypertrophic cardiomyopathy. I would have given it to him right away, because Friedreich's is progressive, and the sooner you do it, the better the outcome, because you don't have permanent damage to your heart. By the time Sean was dilated, he had fibrosis in his heart. He was, it was, you can maybe fix it, but it won't go all the way back to what it was before it was permanently damaged. As long as it was hypertrophic and we weren't getting fibrosis in there, there's every chance he could have gone back to having a normal heart, in my opinion.
Thank you very much. I'm going to remind you again, the phone number for you to call in is 703-844-3231, or you can submit a written comment, and we have a few more written comments to share with you now.
We do. The next one is, my biggest concern is passing away while my children are young and passing away suddenly and unexpectedly. I worry that my ARVC or ACM will progress more rapidly than I hope, and my heart health could worsen.
Wow, that is a big scare for a lot of us with children and families. We don't want to leave anybody, especially before they're grown and able to function on their own. Christy, you're a parent, so I'm going to lean on you here a little bit. As a patient, what does it feel like to have symptoms that you know are life-threatening, and the cascade of the worry for all those who will be traumatized by not only your event, but God forbid, your death?
Yes, it is absolutely terrifying. Because again, I shared that I had 4 major episodes of sustained VT. And 2 of those episodes, I don't know what would have happened if I had not gotten to a hospital to stop me from being shocked over and over and over again by my defibrillator. So this is a situation where defibrillators, again, they're a safety net for so many people, which is wonderful, but it's even not shock anxiety, it's death anxiety, because your heart is doing something terrible, you're being shocked, and if you don't get to a hospital potentially to have stabilizing medications when you're in a shock storm or a VT storm, will you survive? That thought process, that symptom is in the minds of your caller or your commenter, as well as people with arrhythmogenic cardiomyopathy all of the time. And having had that experience again twice, where if I wouldn't have been at the hospital, I probably would not be alive today, is terrifying for me. And it impacts my daily life, and it impacts my daughter's life, because when you'll see from my story later, my dad died in his sleep from the disease. My aunt died of heart failure. So we have this family history already that is terrifying. And then we're trying to figure out, how to live and thrive the best that we can. And again, with the arrhythmogenic community, I'm extremely involved. I'm the chair of the board of SADS. I work with Greg on dilated cardiomyopathy as well, trying to help, but I'm listening to the patients, and they're terrified because the therapies that we have, and we'll talk about this later, but what we have today is not preventing the progression of the diseases. And it's not even potentially saving people from sudden death.
So speaking of the current treatments, Gwen, I'm going to pivot down to you for a second. You and I both shared the experience of being on beta blockers for a really long time in our life, which benefits, but it has its downfall. Can you talk a little bit about the older type of therapies that we've had that weren't actually tested on us, but other heart failure patients, but we got by default?
Right. So Lisa and I, of course, share HCM. I've known each other 20 years. I too have an MYBPC3 gene that was not identified actually until just a few years ago. But when I was diagnosed, the condition was considered grim. There were no real options. They were doing surgical training at the NIH on dogs. There was no really suspicion or any plans at that time. And for many, many years, decades, I would say, we took a combination of drugs, calcium channel blockers, mostly beta blockers, some light antiarrhythmics that were treating more symptoms. So until the first cardiac myosin inhibitor was approved by FDA in April of 2022, the treatment that we received was really like cocktail. Let's try this. If this doesn't work, and is it not working because we're aging? Is it not working because I'm in menopause? I went through menopause in my, mid 40s, 50s, and that was a disaster because you have weight gain, you have night fluctuations, you can't sleep. Is it the medication you need to change? Is it the worsening of the HCM? Is it fluid retention? So that's a special category. So these drugs provided secondary symptomatic relief, but they did not get at the root problem. So we are living in time of more targeted therapies with now 2 cardiac myosin inhibitors on the market. I will say, because I made the personal decision to not have surgery and not have a septal reduction procedure, but to wait for these drugs to be on the market, I began having atrial fibrillation about 7 years ago. Now, AFib is a common side effect of HCM. About 20, 25, maybe 30% patients have AFib. Primarily, it is due from a structural obstruction in your left ventricle. Lisa showed you some of the models there, and that back pressure creates a very enlarged, grossly enlarged is the word they use, left atrium where the signals begin near your pulmonary veins. And because of that stretching and enlargement of your left atrium, even the medications to treat AFib can only be of marginal benefit. So AFib is, for somebody like me who's been symptomatic so long and as I age, was inevitable. So when I actually started on a cardiac myosin inhibitor, what is now in the last 6 or 7 years has become a Rubik's Cube, I call it. Is it the HCM? Is it the AFib? Is it the side effects of new medication, which, let's face it, any first-in-class drug put into real-world evidence is going to come about with a lot of unknowns. So those three things now require a management that is part guessing, part fact, part science. And I'm very optimistic that I am now going to get care continually. But the last couple of years, it has really been challenging. At one point, I was in persistent AFib for 18 months. And the question really in everyone's mind was, is this a secondary side effect of the cardiac myosin inhibitor, or is it a worsening of my underlying condition, or how to actually go about treating it? And I'm still going through that. I've been cardioverted 4 times this year. I have my 3rd ablation next month, and most likely will switch to a different cardiac myosin inhibitor to give it a try. So even for HCM patients, we are so fortunate that there are options. I mean, having been diagnosed in an era when there were no options and told you will have a short life, it is exciting and rewarding and just incredibly phenomenal that there have been guidelines and targeted therapies, and that pharmaceutical companies are devoted to this condition. And so I will say, even though I'm sort of on the back nine of my experience with HCM, it's quite encouraging that in the future there will be more targeted therapies that really bring either an end to this condition or a management that's very much consistent with the normal life.
Thank you, Gwen. We've got about 10 more minutes in this particular segment. We're going to be taking a short break at the end of this segment, and we're going to be coming back to talk about the upcoming therapies, and we're going to hear again from Dr. Seidman on where the science is evolving. So we do hope you stay with us for that. I do want to pop over to Giselle for just a moment. And Giselle, when we've spoken previously and you speak of your father with such love and admiration, if he were here today, what do you think he would think about not only your advocacy, but the disease state, where it is today versus where it was when he was diagnosed? And what symptoms would he maybe have gotten over if he had the more targeted therapies?
His humbleness is what was the issue when he was around. He often said to me, I just want to know what's wrong with my heart. At that time, amyloidosis was still on the back burner because there were no treatments available when he got diagnosed. And the same year he passed, I got diagnosed. There were still no treatments available. So my dad was proud of me being his only child. I got my bachelor's degree, and then 20 years, I got my master's degree. But the important thing now is I went back and became a certified health and wellness coach so that people don't have to go years of trying to figure out what's wrong with them. I'm an advocate for this disease. I speak to people all over the country about it. And I think he would say, job well done. He was a very passionate person, and an example of his kindness, he made sure every one of his 4 sisters received a Valentine's gift every year. He sent them Mother's Day cards every year. So I've picked up that characteristic from him, and I have a passion for trying to help people no matter what. And being able to help people push through the monotony of trying to figure out what's wrong. These symptoms for amyloidosis is like putting together a puzzle. You got so many different caveats to it. And until you get to a provider that's able to put those pieces together, you'll still be trying to figure it out. So I think both mom and dad, they're back together again, and they're proud. I know they're proud of me. Trying to advocate for other patients that may have this disease. I met people with heart disease that didn't have amyloidosis, but it had to be tested to find out and rule it out.
Absolutely.
So I am so happy to be a part of the community and be able to advocate for other patients with this disease.
We're so happy that you're here with us. We do have another comment coming in from the Danon community. We'd love to hear from them.
Yes. So it says 20% of my heart is fibrotic due to Danon's disease, putting me at constant risk of fatal arrhythmia and sudden cardiac arrest. If an innovative curative therapy does not become available, and for women, I will likely be in heart failure within the next several years and needing a heart transplant. Thoughts of either sudden cardiac arrest or death, sudden cardiac death, or drawn-out road to heart failure are both equally terrifying and prevent me from being present with my young children and family. My children are also at 50% risk of inheriting my mutation, putting their own futures at risk. We need transformative therapies for both males and females with inherited cardiomyopathies so that our lives don't revolve around specialist appointments, aren't filled with constant fear, and aren't limited by the mutations.
Wow. One of our community members we worked with at HCMA before Danon's Foundation was organized. The son was transplanted about 5 years ago, and the mother went for transplant just a few weeks ago, and unfortunately it wasn't successful and she passed. There aren't good options for these families, and these deaths they just keep happening. They're not high numbers, and maybe they don't make the front page of the news or they don't go viral, but they are catastrophic events to these families, and we really do need to focus on all of those with disease, not males, not females, but all. So we know that the young males, you saw the heart earlier, are severely impacted with mass hypertrophy, but the women are impacted as well, and we need to have a better understanding of them. So shout out to all the women with Danon. Now we have about 4 more minutes, so I'm going to do a round robin. You each have a minute left to talk to our friends at the FDA. I'm going to start with Gretchen, then go to Christy. So Gretchen, final comments.
So Friedreich's ataxia is a recessive disorder. And the heart, for people looking at it, the flashy part is the ataxia. And I don't mean that in a good way. That's what people see. And I'm not saying it isn't bad, because it is very bad. There are a lot of bad things about that. But the heart is what kills them. And it's because they don't make enough of a good protein. It's not even that they make a bad protein that's destroying their bodies. They just don't make enough of a good protein. And Sean was optimized on every medicine that he could be on for his heart, and it still wasn't enough. And so gene therapy, it doesn't even have to be perfect if it gets to the heart. If you can get it to a carrier status, I make about 40 to 60% of what a non-carrier makes of frataxin. If you can get it to that state, you fixed it. So I really feel like gene therapy is the way to go with this.
Thank you, Gretchen. Christy?
Sorry, thank you. There's so much I'd like to say. I think, first of all, I also run my own support group for desmoplakin cardiomyopathy, and I have about 600 and so people in that group. And I talk to them all of the time. And as a community, they're terrified, right? We're terrified because these are young people very often who've had sudden cardiac arrest, or someone in their community has died. And now they have to manage arrhythmias and also heart failure symptoms. So they're dealing with two sides of a coin that are both very, very difficult. And there's not good options for them. So we are appreciative, of course, of the heart failure medications that are out there that help prolong ejection fraction and those things, and the antiarrhythmics and the ICDs, but it's not curing the disease, and it's not stopping progression of the disease. It's almost like Band-Aids. And again, I know we were a long way from when my dad passed, but there's still such a long way to go. And I just really hope that we can get good solutions. And again, for desmoplakin, the adenovirus component isn't going to work because the gene's too long. And there's so many of the other genes that are like that. But there's other therapies that are on the back burner that I know are people are trying to solve that problem. And I'm so excited about some of the other solutions I've been hearing about. So I just really hope that those will come to fruition and solve the problem. I would love it in my lifetime. Thank you.
I've been told I have 1 minute to go, so I'm going to go
sorry.
to, that's okay. I'm going to go to Greg, and that'll be our final comment for today. Greg?
Great. Thank you again, FDA, for listening. There's 1.5 million people with genetic cardiomyopathy in the United States. They, as you hear, live sick for a long time, get a transplant, or die. And there's very little hope. Our hope is in genetic therapies, albeit AAV, RNA, CRISPR, or a combination thereof. And I also believe with, not only saving lives and a lot of misery with these potential gene therapies, given all the costs, when for cardioversions this year, I believe that gene therapy will also save the taxpayer and save our healthcare system money. So thank you. And this is really, really important. And I think also can help our country and the healthcare system financially. Thank you.
Thank you all. I'm sorry, Giselle and Gwen, we didn't have final comments from you, but you can submit them in your written comments and it'll be in the Voice of the Patient report. Thank you all for joining us for this segment. We're going to take a brief break. We'll be back in 15 minutes with Dr. Christine Seidman talking about future therapies. Please join us then. And remember, you can call us in the second segment to provide comment at 703-844-3231. Thank you so much for joining the first segment. We look forward to seeing you in a few minutes. Welcome back to the second segment of our externally led patient-focused drug development meeting focusing on genetic cardiomyopathies. We're going to hear once again from Dr. Christine Seidman, from the director of the Cardiovascular Genetics Lab at Harvard Medical School, about future therapies in genetic cardiomyopathies. Take it away, Cricket.
Well, welcome back, and I'm delighted to tell you now about some of the opportunities for the development of novel therapies and the impact that they may have on our patients. I've told you previously that there are many genetic causes of cardiomyopathies, and I think this picture illustrates how many there really are. I'm showing you on the left the inside of a cell, all of the different molecules that need to participate for contractility and relaxation, and on the right, two major classifications, hypertrophy and dilatation, that occurs from cardiomyopathies. And then I hope you can see that under each of the headers of the different types of hypertrophy, such as amyloidosis, Danon disease, and hypertrophic cardiomyopathy, there are molecules that are listed, gene names rather, that are listed. And these are the known genetic causes of each of those phenotypes. We now know that there are hundreds of individual genes that cause genetic cardiomyopathy and literally thousands of different mutations. These are largely in an individual or in that individual and his family members, and they are different between other families that are not genetically related. Despite this incredible complexity, or what we call genetic heterogeneity of cardiomyopathies, we recognize that these cause disease by two predominant mechanisms. In one, we call it dominant negative, and what that means that the mutation causes a change in the DNA that alters the protein structure. A single amino acid is substituted for another, and that can alter the function of the protein, either increase its function or decrease its function. But the total amount of protein that is made by that mutation is normal. And since we have two copies of every gene in our body, 50% of the protein would be abnormal, 50% of the protein would be normal. On the right, I'm showing you what happens with haploinsufficiency. This is the other consequence of cardiomyopathies. And in this instance, one of the two gene copies is shut down. It is inactivated because of the mutation. Now that protein level is reduced by 50%, but all of the protein that is made is coming from the normal gene copy, and so it has normal protein function. What we recognize from understanding these genetic mechanisms is that there are a variety of strategies that could address them, and they would be quite distinct in what they aim to do. For dominant negative ones, if you have abnormal protein that is present as well as normal protein that is present, you might want to just silent the mutant protein. And we can do that at the DNA or at the RNA level. Alternatively, if you're not making enough of a protein because you only have one gene copy, you might want to deliver exogenous protein or a gene that could then make the insufficient amount of the protein. And in both of these instances, if you had the capacity to directly correct the DNA sequence, you could cure each of these different causes. Now, there are several strategies that are being used from these genetic approaches. One of them we call non-durable because they do not target the DNA, which is fixed in every cell and does not change over the life of that cell, but rather the RNA or the protein that is made. And other therapies are more durable, and those actually alter the DNA either to correct it or to silence it or to modify it in another way. And we're going to go into those mechanisms a bit more on the next slide. So this is a schematic of how we would deal with insufficient protein because one gene copy is inactivated by a mutation. And what we could do would be to take the exogenous gene, or what we call a copy of that gene, or cDNA, and put it into a vector as shown in that hexagonal structure on the top right of your screen, and deliver that through the bloodstream such that it would go to every cell in every tissue in the body, or more specifically, be expressed only in cells in which we put sequences so that the structure inside of the vector is only made in cells that need the protein. There are a variety of different genes that we know could benefit from this type of therapies, including the one shown at the bottom of the slide. A number of different muscular dystrophies, Friedreich's ataxia, Danon disease, as well as arrhythmic cardiomyopathies, and some forms of amyloidosis. Now, while these are beneficial opportunities, the problem is that they're not durable. We don't yet know how long these vectors will hang around in a cell and how much protein they will make, whether too much, too little, or whether it'll be adequate but unable to be regulated the way the normal protein is made. Moreover, we know that many genes that are mutated in cardiomyopathies are very, very big, and we simply cannot produce them into a vector and deliver them into patients. And for these, we would have to minimize in some way that particular protein or gene, and that may alter the function of it. So, if we move forward, another opportunity that is addressing the dominant negative mutations, and again, these are ones where the protein level is normal, but half of the protein is functionally abnormally. Here, what we could do would be to silence the abnormal RNA or silence the abnormal gene, and we can do that in a variety of different ways, as shown here. Some of the techniques are called a small interfering RNA, a microRNA, or an antisense oligonucleotide. And while these are effective and can sometimes be delivered without a vector, such as the antisense oligonucleotides, they are not a total solution. Because, first of all, we know that they can only be used when the residual gene is functional and can maintain the normal properties that two copies of the gene could do in most of us. So it's limited in terms of those genes would be appropriate. Second of all, we know that the uptake of some of these molecules is fairly broad, and we don't yet fully understand if there might be consequences to delivering these important and very biologically active molecules into cells that don't normally make the gene that we're interested in. That being said, these, because they are transiently delivered and over time are lost, allow us to test things and see if they are safe and effective, at least for a period of time. And they are under clinical trials now in a variety of different conditions that are shown at the bottom of the slide. I thought it would be helpful for you to see how we use these in a research laboratory. This is a mouse model of hypertrophic cardiomyopathy shown on the left, and I hope that you can see that the heart structure shown on the bottom right panel, on the left panel, but the right side, shows brilliant blue, which is due to fibrosis accumulating in the heart. The walls of that heart are also thickened. And we look to the left of that, we see that the orientation of those cells are very abnormal. When we add a mutant RNA that we silence with our ASOs or potentially with an siRNA, we can prevent the abnormal protein from being produced because the RNA is no longer present in that cell. And in those animals, when we look at their heart structure, it's perfectly normal. There's no thickening and there's no blue material suggestive of fibrosis. Moreover, the structural organization of the heart is preserved. So that's well and good, but there are limitations to that. And so people have begun to think of more durable therapies in which we could directly affect the DNA that is altered. And current approaches to that are really using molecules that are called base editors. These are very important and highly dynamic molecules that allow us to change one nucleotide into another nucleotide without altering the structure of two strands of DNA, something that has not been feasible until the past 5 to 10 years now. Base editors have the specificity to target two different bases, an adenine or a cytosine, and they can convert each of those nucleotides into one different nucleotide, an adenine into a thymidine, a cytosine into an adenine. Now, while these have the opportunity to make changes that could potentially correct or permanently silence DNA, there are a number of limitations. For example, they would only be applicable to diseases where these two nucleotides are the cause of the cardiomyopathy. They have only a few components, but each of those components are quite large. And at present, we can't deliver them with a single vector, but we need at least two. That actually can allow for in vivo activity and to prevent a disease from occurring, but at present has not yet been shown to cure a disease and revert the tissue back to normal structure after a disease has established itself. That being said, I wanted to point out to you that this has already been used in rare instances in a first in human approach to treating a rare genetic disease, in which a child who was born with a devastating and lethal condition received gene editing using base editors that was able to correct the genetic disorder within the first few months of life and at present appears to be doing quite well. We've actually tried this approach in our mouse models of hypertrophic cardiomyopathy, and I want to show you a little of those early results. On the left panel, you'll see that the normal sequence is altered from the mutated sequence by one nucleotide, which substitutes one amino acid for another. That substitution allowed us to use an adenine base editors. And when we treated our mice and then sequenced them, we found that delivery of our base editors resulted in over 90% of normal sequences being restored. The clinical consequence in the mice are shown on the right panel. In the untreated animals, shown in red, they develop hypertrophy. In the treated animals, shown in blue, they, like the control animals, have a normal thickness of their walls. And when you look at the heart structure and the accumulation of fibrosis, similar to what I showed you previously, that those that had editing have normal structure of their heart, normal cellular organization, the untreated do not. Well, that sounds like a golden opportunity to advance towards translation, but unfortunately, there remain significant limitations. These include, again, the need for two vectors to deliver, which will raise the amount of viral load if we use a viral vector, and that alone has consequences. There's the potential for off target editing and effects. And as I mentioned before, one can only edit specific bases. And for many genes that are mutated and cause cardiomyopathy, those two nucleotides are not altered. Most importantly, these kind of approaches can be specific for one patient, but are not going to address the hundreds of different mutations because each of them would have to be designed for every single patient, and that simply will not be feasible. The newest approach to gene therapy is using something called prime editors. And these are derivatives of base editors, but far more complicated and advanced. Unlike base editors that change only two different nucleotides, these have the capacity to change many nucleotide, one to any different base pair, a series in a row, or to insert new sequences or to delete them. Like base editors, they don't have the adverse consequences of double stranding breaks in the DNA. And despite their possibilities to deliver really life saving therapy and versatility, they unfortunately are too large to deliver in our simple, novel, in our vectors available today, and will require the development of new and improved vectors. They also have limited uptake by cells, and that, in fact, has limited their opportunity for immediate translation. Despite the progress that's been made in these genetic therapies, there's a lot more that needs to be done. We truly need to know the genetic cause of each and every patient's cardiomyopathy to understand the mechanisms by which they cause disease and thereby to establish the best genetic therapy for those individuals. We need to know which are the right patients to treat and when is the right time to treat them. We also need to advance the development of vectors to deliver these new and potent therapies. And as we move from mouse models and preclinical trials to human data, we need to have full disclosure of the safety, the efficacy, the durability, and the good news and bad news, the off target effects, and those that are intended. Most importantly, education of our patients, our doctors, our regulators, and the public at large need to fully understand the risks and the potential benefit for our patients with cardiomyopathy. And as we get consent for patients to participate, these need to be fully transparent, clearly articulated so that they realize how they are contributing to the future of medicines to hopefully benefit all and the risks that will come with that opportunity. I thank you for your attention, and I look forward to hearing more from our patients about these opportunities.
Thank you, Dr. Seidman, for those inspiring and aspirational projects that are already underway and the hope for so many patients with these new technologies. We are now going to hear from some more patients experiencing cardiomyopathy. Now we're going to shoot over to Becca, and she's going to tell us about her family.
Hi, my name is Rebecca Lang. My family is from Chelsea, Michigan, and we are here representing people who have Friedreich's ataxia. In our family, my daughter, Arlie, who is 9, has Friedreich's ataxia, and my son, Abel, who is 5, has Friedreich's ataxia. So Arlie was about 2 years old when we first started noticing that something wasn't right. She was tripping, she was struggling to run and to jump. Her gross motor skills were delayed. And so we went through testing of scoliosis, and we found out that she had scoliosis. From there, we were recommended to get an echocardiogram. And her first echocardiogram showed that she had HCM. And so that was interesting because now we had these 2 different diagnoses, but she was still having these neurological issues. From there, it took us about another 6 months to a year to finally figure out that she had Friedreich's ataxia. It was a very long, slow process to get everything figured out, and many hours online and researching all throughout the night. My son Abel is currently 5, and we found out that he had Friedreich's ataxia after we found out about Arlie. So when Arlie was diagnosed, our family decided to get the rest of our children tested. My daughter Annie is not a carrier of anything, but we found out that Abel had Friedreich's ataxia, and he had just turned 2 years old. So he was still just a little toddler, and it was devastating. At his first echo, he did not have any cardiac complications. And as he grew up a little bit, his EKG did start to show some abnormalities. And as of now, he just now is at 5 years old, is starting to show some neuromuscular issues. So as a mom with 2 kids and one without some significant health issues, we have a lot of worries, big and small. And a lot of them are similar to just the same worries that every parent has, like bedtime and getting places on time. But coupled with that, we have things like we also have to be our kids' advocate and their physical therapist and their occupational therapist. And we struggle with bedtime, especially for Arlie, who has to wear back braces and feet braces. And she struggles with sleep apnea and heart issues while she sleeps. So there's the normal bedtime routine on top of all of that she has to go to. And she is my spicy kid, and she does not want to wear a back brace and feet braces to bed. And I don't blame her because they're highly uncomfortable. But at the same time, we have to push past her tears and her anger and make her wear these so that she doesn't have to have surgery in a few years down the road, which will cause major complications with her heart. And so just that daily kind of wear and tear on her and on us is really hard. And at the same time, then we worry about my, Abel, who's coming up. Is he going to have to go through these exact same things? And just dealing with all of that. And now Abel is old enough to see what his big sister's going through, who also has Friedreich's ataxia. So we have the emotional side for him where he's asking, am I going to be in a wheelchair? Am I going to have to wear a heart monitor? Am I going to have to do these things? And she is just struggling with how unfair it is for her to have to do this while her sister, who is younger, does not. And my sweet Annie, who doesn't have anything, has 2 siblings who have all of these issues, and she kind of has to take that in stride. So we worry all about if we're doing the right thing as parents, as well as some things like when Arlie was in a play, she wanted to go and she loved being there, but we couldn't leave her there because we were worried about her heart, and the play was a very long time for her to practice. She refused to use her wheelchair and her walker, and she would fall during the play in practice. And so we had the constant worry of, is she going to hurt herself, or she's going to break a bone? Is she going to fall and hurt her head? Is her activity level too high for her heart to manage? So those are some worries, some small things that seem small but are huge for her, is just being left out. And so we kind of have to balance going to things and going to birthday parties and going out with our cousins and how often we see people, because she wants to play and she wants to run and she wants to do all of those things. But when we're with a big group of people, she can't run with her cousins. So they all take off running and she is left alone again. And so we have this constant ebb and flow of how do we make sure her life is extremely enjoyable, but to try and take away those times where she just truly gets left out, not to her own fault. My hope for my kids is that they get to grow and learn and love and make their own choices in this life. I hope that the clinical trials of genetic therapies are able to provide safety for their heart and their whole bodies. And we're just really excited for where this is going. And my hope is that as these therapies become available, families have the decision and the availability to pick the trials as soon as possible instead of waiting until a kid may be 18. As far as my daughter's concerned, she might not make it to 18 to where her age is available to get her in this trial or any trial. So having the option to get into a genetic-based therapy trial as soon as possible could truly save these kids' lives.
Hi, I'm Christy Johnson, and I live in Scottsdale, Arizona. In 2022, I went into sudden ventricular tachycardia just standing at my kitchen counter and was diagnosed later that year with arrhythmogenic cardiomyopathy, the desmoplakin gene variant of that. I was 47 years old at the time. It came out of the clear blue sky until later we put family history together and realized that my father, who had passed away in his sleep at age 52, was very likely positive. And my aunt, who died of heart failure after battling it for 20 years, was also very likely positive. We know this because my cousin, I have 5 cousins that are positive that are related to my aunt. And unfortunately, my daughter also is positive for the desmoplakin cardiomyopathy gene. So to start my journey with this disease, I went into sustained ventricular tachycardia, and in the hospital, they had to shock me several times over several hours to get me out of sustained VT. They also had to use IV medications for that. It was extremely traumatic because I was awake for the first shock. I felt every bit of it. And it's being shocked, either whether it's somebody in the hospital shocking you or by your defibrillator, which I now have, is really hard to describe, but it is like a donkey kicking you in the chest. Since that first episode, I've had 3 more episodes of shocks. The first one was in October of 2021. I was just at a children's party, right? Having fun with kids in a zombie apocalypse game. Wasn't even running around, but got shocked because of the adrenaline that went to my heart. And again, I thought the zombie actually hit me with a rock, but it was so painful that I stopped the entire game, obviously talked to my doctor, and I had been in sustained VT. In April of 22, I was just sitting at a quiet restaurant with my husband, and out of the clear blue sky, I was shocked twice. I was afraid to move. I was afraid to breathe because I thought I would continue to be shocked. Went to the hospital, spent several days there. And then in July of 2022, I was shocked 10 times in a row while at a friend's birthday party. So again, just sitting in a restaurant with my friends, we had just sat down, everybody was celebrating, and bam, a wild scream came out of my body and everyone stared at me, and it was so painful. I got taken in a car, started to drive toward a hospital, and had 9 other shocks over the next hour and a half until I could be stabilized. So this diagnosis has impacted my everyday life significantly. In terms of symptoms, unfortunately, I'm dizzy most of the time, very lightheaded. I kind of feel like I walk like a drunken sailor some of the time, which is, attractive. But beyond that symptom, unfortunately, I have a lot of fear and anxiety that's developed from these episodes of being shocked. Sometimes I'll get a social invitation, a birthday party invitation, and I'll have to really think about, do I want to go to this? Because it's going to be anxiety provoking for me. And, a lot of my episodes happened in social situations or in situations where there was adrenaline and even joy. So is this something I really want to do? And that's really hard because I am a very social person, and it's really impacted my life quite a lot by that. And I've had a lot of therapy as well to address these issues. I also feel a lot of palpitations, and the palpitations actually increase the anxiety, which then increases the palpitations. So that is an everyday event for me. And I've had to work a lot again on recognizing that fear is a thing and that's okay, and try to talk myself out of it. But it's the anxiety and the fear with this condition is beyond anything that's really easy to explain. I've had lots of guideline-directed therapies for this disease. I've been on all of the medications, and they make me tired and dizzy. I've had an epicardial ablation, which helped, but I had so many side effects and had to go back in for 2 more procedures. I've had 2 different ICDs, which have saved my life, but also caused complications too. I've got, backlash in my heart. I've got other things going on because of the ICDs. We need more different therapies. We need therapies, genetic ones, hopefully, that can be a cure for us, and not just a Band-Aid, and not just something that is temporary, because my disease is still progressing right now. My daughter, I want her to have the future that she deserves. She's in medical school right now, and she wants to help others. But if things go the way they are, she may not be able to pursue her entire life in the thing that she's most passionate about, which is helping others. I am actually the chairman of the board of the SADS Foundation, and I am passionate about people with desmoplakin cardiomyopathy as well. I run my own support group. And I cannot tell you how many people really are struggling and suffering and need your help to please allow additional therapies to come through and allow us to find a cure. Focus on those rare diseases. Focus on the things that really matter to people who are trying to make a difference in this world. Thank you.
Hello, everyone. My name is Scott Popchus. Thank you for the opportunity to come and speak to you today. 6 years ago, I had a similar opportunity. Let's take a trip back in time and hear a bit about my story. I was first diagnosed with hypertrophic cardiomyopathy in 1990 when I was 23, following my father's diagnosis with HCM. For the first 5 years, I remained mostly asymptomatic, but in 1995, I experienced my first noticeable symptoms, a type of cardiac arrhythmia called supraventricular tachycardia. Since then, there's been nothing simple about my journey with HCM. I've had shortness of breath, fatigue, various types of arrhythmia, both atrial and ventricular, and in recent years, issues related to heart failure. I watched as my father's condition worsened, eventually requiring a heart transplant. I have been treated with various medications, had multiple radiofrequency ablations and cardioversions, a septal myectomy, and an implantable cardioverter defibrillator, the first one in 1999. While all of these treatments have been helpful, it's because of the ICD technology that I'm alive today. Many of the medications I've used come with side effects that have ranged from annoying and disruptive to intolerable and toxic. The fatigue and brain fog from beta blockers make it difficult to focus or get through the day without taking a nap. There are periodic blood and pulmonary tests to make sure the antiarrhythmics, specifically amiodarone, wasn't harming my liver, thyroid, and lungs. And there's the fear of starting new antiarrhythmics, like sotalol, that can provoke the very arrhythmias they are supposed to control. The Lasix makes my long LA commute challenging, to say the least, and disrupts my daily work routine at the office with frequent and embarrassing trips to the restroom. The benefit of living to see another day far outweighs the difficulties, challenges, and speed bumps along the way. Looking to the future, my hope is that technological innovations will continue, and leads will become more durable, devices will get smaller, batteries will have a longer life or can be recharged, and communication between a patient's device and their physicians will be more mobile and accessible. And finally, effective drugs with less serious side effects that are specifically targeted at HCM and its complications will be developed. Thank you. Now, if you excuse me, I have to hit the restroom. I'm back. And I brought a friend. Just kidding. Well, not really. My friend here stays well hidden in my car. In the event the diuretics kick in and I can't find a restroom or can't get to a restroom. You've heard of people with electric cars having something called range anxiety, which is they can't find a charger, or will they have enough range to go where they need to go? Well, I was having pee anxiety, and my friend came along and helped alleviate that. Ladies, I wish I had a suggestion for you. I'm sorry, I don't. The past 6 years have continued to present challenges to me with HCM. I've experienced more arrhythmias, both ventricular and atrial. I've had ICD shocks to correct dangerous rhythms. I've had lead failures, which required extractions and replacement. And I've continued my battle with heart failure, which is an ongoing daily struggle, frankly. Monitoring fluid intakes, monitoring salt intake, monitoring your weight, adjusting diuretics. But fortunately, I have a phenomenal team that oversees my care and works with me hand in hand as I walk through this. In recent conversations with the team, the idea of evaluating for transplant has come up. So now I know that may be on my horizon at some point. I got to be honest with you, HCM sucks. You know, when I first got this disease, it was barely noticeable. It didn't really limit me in many ways at all. But as time has gone on, it's declined and the symptoms have worsened. And it has limited me. And I can't do some of the things I used to do that I still want to do. Thank God for my e-bike, which is a godsend, by the way. I can go out and ride my bike, and if I hit a hill, I just add a little electric in and it helps me pedal better and get up the hill. So that hasn't been taken away. But it's rough. It's rough. You know, HCM is a disease that affects the physical, it affects the mental, and it affects the emotional side of a person. But not only that, it affects the people around that person, the family, the caregiver. It's tough on everyone for different reasons and in different ways.
I wish there was a way to eradicate HCM. I wish there was a way to prevent it from developing in people. I wish there was a way to cure it. But I go on one day at a time and do my best. And I just want to encourage all of you to continue doing what you're doing. Continue working, continue researching, continue pushing the edge in ways that could help patients like me in my future and people like my kids and grandkids who may face this as well. So thanks for your time, and I appreciate it.
My father died at age 52. I was 14 when he got sick. And the diagnosis was cardiomyopathy. At that time, it was in 1976, they thought it was a post-viral cardiomyopathy. And that's what we thought until my brother dropped dead at age 35 while water skiing. Some years later, we figured out that our family has RBM20. I didn't feel well. I had progressive symptoms of heart failure. I'd wake up in the middle of the night, like a lot of patients with cardiomyopathy, feeling like I was being waterboarded, like I was drowning. And then, you know, our family's disease is one that is prone to sudden deaths and cardiac arrest. So when my brother Rich died, got on a treadmill, had arrhythmias, they basically said, you know, you're at a very high risk for having a sudden death, and we need you to get one of these new implantable defibrillators that had just been invented at Hopkins. I happened to be in surgical training at Hopkins. We thought it was going to be the end of my surgical career, but that worked out. They thought I was the first surgeon in the world to get a defibrillator, and they thought that some of the electronics in the operating room would interfere with my defibrillator and make it go off. So once we tested all that and I got cleared to go back to my training program, I finished that and became a transplant surgeon. And then I'd say like the next 30 years were periods of relative stability punctuated by near-death experiences. And I had 7 cardiac arrests before, you know, I was sick enough to get listed for a heart transplant. And after I woke up from my heart transplant, I dedicated the rest of my life to that cause. And so now we're in the first clinical trials for xenotransplantation using gene-edited pig organs into humans. We're starting with kidneys, but the hope is that in the not too distant future, we'll be doing hearts as well. Beyond that, then there's curing the diseases, the genetic diseases that underlie many forms of cardiomyopathy. And, you know, that's going to be done. We have the technology now. I think it's just a matter really of deploying it, you know, in the right settings, figuring out how to get CRISPR and, you know, other gene editing technology in the right place to really restore healthy heart function. Yeah, I think, you know, there's a lot of different genetic approaches, and RNA-based therapies are, you know, one of them that I think are demonstrating a lot of hope. I mean, some of these genetic disorders are just, you know, one base pair that is mutated and results in one amino acid change, which then, you know, makes a gene not functional and causes cardiomyopathy. That's certainly the case in our family. They're targetable. They, you know, these kinds of genetic disorders, you know, we have the ability to make gene edits. It's really a problem right now of delivery, how to get them to the right place safely. And you know, this is the future that we need to embrace. We need to be daring. We need to take some risks. I think if you talk to most people who have this disease, have these diseases, they are willing and really would embrace any kind of hope, any opportunity to improve their own lives and the lives of their family members. And so we really need to move this forward. The treatment of rare diseases is such a critical thing, you know, that has to be addressed. And you know, again, it has such a profound effect on people who are afflicted by all these different rare disorders. It really needs our attention. It really needs both funding and you know, clinical trials, and really, you know, greenlighting these opportunities to really change the future in rare disorders. Normally we think about life-threatening diseases as our own individual diseases, but these are family diseases. It's a very different thing. And you know, there's parents who have passed these diseases on to their children, and that's a devastating feeling. And that feeling of responsibility, you know, for their well-being in a different kind of way that, you know, we normally think about that when we're parenting. And, you know, having to really think about not just your own health and how you're going to get through this, but, you know, every affected member of your family, and then the next generation after that. It weighs very heavily. I can't even really describe, unless you have experienced that level of tragedy and sense of, you know, impending problems. It's a difficult thing to really even explain what that feels like. So my name is Robert Montgomery, and I'm a transplant survivor. I'm now 7.5 years out from my heart transplant. And I'm also a surgeon, a transplant surgeon, and I've done thousands of transplants and conducted a lot of research to try to advance transplantation, to try to increase the number of organs that are available through things like xenotransplantation, and also making living donation more accessible. And I just want to thank everyone who is involved in this process, and really tell the families who have been affected by tragedies and by the fear of genetic disorders that there's hope, and there's going to be a brighter future.
As you've just heard, we come from all walks of life in the cardiomyopathy community, and our sincere thanks to Dr. Montgomery for sharing some of his time with us after he was named Times 100 of last year. So thank you so much for all of our participants who shared their stories with us, and it gave us a lot to think about the future. We are going to pop over to Joey right now for some more survey results. Joey, off to you.
All right, Lisa, thank you. Let's jump back into some of the questions we ask our survey participants. Well, the question here, what heart therapies have you or the affected person used in the last 10 years? And you can see that blue bar across the top, just over 85% beta blockers. Under that we're talking about that green line that goes across now, about 56% said device therapy. So That's talking about implantable cardiac defibrillators, ICDs, sometimes they call them, or SICD. You see nearly 40% dealing with diuretics, and 39% blood thinners. Will also give a mention of about 32% with calcium channel blockers. The question then we ask, what are you currently doing to manage the cardiac health or heart symptoms beyond just these therapies for your heart muscle disorder? So let's look at that yellow line across the top, and you can see that about 65% of the respondents are following the recommendations of their heart care provider to seek some advanced therapies. Then under that, about 60% say lifestyle modifications, and these are things that they're doing at home, work, exercise. Under that, we've got nearly 50% that are changing their dietary habits and staying hydrated, and about 50% that are looking at weight maintenance. Another question we ask, have you wanted to participate in a clinical trial with an experimental treatment for heart muscle disorder? Look at that green line across the top. Nearly 50% said no, they have never been asked or informed of one. About 17% said yeah, they had tried to enroll in a trial, but they did not qualify. You can see the different colors in the explanations on down the line. Let's hit a couple more. About 15% say no, but they are interested in a clinical trial. And about 14% said yes, and they are enrolled in a trial. So another question for our respondents, for those impacted, how much in general have these treatments and lifestyle choices helped improve your quality of life? We can see for about 4% that there was no benefit at all. But look at that blue line. About 34% said, yeah, they helped somewhat. About nearly 30% said it helped a lot. About 26% said a very significant benefit. 7% were not sure how to answer. So let's look at one more thing here. Considering your affected person's current health status, which symptom do you most want a future drug or treatment to eliminate or improve? We're going to look at the top 4 results that we have from all of the different responses that we received. About 44% say that one of the symptoms that they would like to eliminate is the worry and risk of sudden cardiac arrest. About 43% worried about advanced heart failure and would love to work on that. About 35% say shortness of breath. So there's another common thread we hear coming up throughout this survey. And about 35% say looking at arrhythmias or heart palpitations. And the last one here we're going to look at is a top 3. The question, let me read that to you. Have you participated or wanted to participate in a clinical trial with an experimental treatment for heart muscle disorder? So let's look at the top 3 responses we have here, and you can see on the side there those red bars that go up. 65% of respondents said how the treatment might improve their health. We look at 64% who have concern over risk of serious side effects or serious side effects, adverse reactions of participating, and up to including death. And then about 40% of those, the proximity of having to travel to participate in that study. So thank you to those who participated and gave us those results. We see how those play out with what folks are saying today and hearing from these Zoom participants. Lisa, back over to you.
Thank you so much. And I'm going to pivot right over to Genevieve, who's going to talk about what is coming up in our next session and what we'll be discussing with our next panel.
Thanks, Lisa. So a few of the questions we'll be discussing is what are you currently doing to manage you or your loved one's genetic cardiomyopathy symptoms? How effectively do these treatments address the most significant symptoms and health effects of you or your loved one's genetic cardiomyopathy? What are the most significant downsides to your loved one's current treatments, and how do they affect daily life? And short of a complete cure, what specific things would you look for in new treatments for genetic cardiomyopathy? What factors would be important in deciding whether to participate in a new research trial? And we're looking forward to hearing from our Zoom panel.
And you're able to call in if you'd like to give comment on this particular session at 1-703-844-3231, or use the comment form and you can submit your comment that way. And please remember to take the survey after the session. So we're going to bring up our next panel, I believe. There they are. Look at all these lovely ladies today. Hello, everybody. Thank you so much for joining us. And some of you have already shown up in some of the video vignettes and some of you have not. So we're going to take a moment to hear from some of those names that we haven't seen yet on screen. And Fida, I'm going to start with you. If you could give us just a very quick understanding of the very complicated family history that you and your family have, and that'll set me up for my next question.
Sounds great. Thank you so much for having us. This is a great opportunity for us to be here. Very, very excited to be here. So let me just start by telling you a little bit how savagely HCM has attacked my family, and for some reason, the female part of the family. So I start by saying that HCM has been generational in my family. So my mom was adopted, but as far as we go back, I have an aunt that died from HCM at the age less than 30. And then I lost my sister. Also, she was less than 30 years old because of a sudden cardiac arrest right in front of me. And then we did genetic testing, and due to my daughter passing out at school, she was 10 at the time, only to discover we have a genetic mutation in our family. It's the TNNI3, which made, I have twin daughters, so both of them had to have an ICD, including myself. But then it didn't stop there. One of my daughters, her name is Ella, she ended up struggling the most with HCM. She spent between the age of 10 years old until 17 years old, extreme multiple ICD shocks to bring her back to life. She maxed out on her medication, and the last resort was when a big that happened to her, and the ICD had to fire up multiple times to restart her heart. And she was 16, 17 at the time, and that's when we reached a roadblock. And her team had told us her only resort now to stay alive is a heart transplant. And she went through the heart transplant, and I'm very grateful for that. She's in college right now, and honestly, I didn't think I would live to the day to see her going to college. I didn't think it was possible. So with that, my other daughter also, she has a heart, the ICD. I have the ICD. I would like to extend a little bit more about, I want to say I'm very grateful for having the pills that we had to manage the symptoms of HCM. I am so grateful for that little device, the ICD, that helped my daughter's life, saved her life so many times. So grateful for this. But I feel like when I look back at my family and how other families, while you think my family maybe has gone through so much, like a lot of tragic events, death, transplant, all that, unfortunately, my family is not that special or unique. In our HCM community, well, my family is normal. There's so many families like mine who lost a lot of people to death, transplant. And then let's not forget, the effect of all those ICD shocks and this traumatic events that we live through, and how it give us PTSDs and high anxiety. So while we have those treatments, which we are grateful for, we pay a high price. We are still paying the high price for having the disease and dealing with it on a daily basis. And for this reason, I I'm not ungrateful for what we have. I just don't think we have enough, and I just want more for my family. I don't want to see my other daughter going through a heart transplant. And if my daughters were blessed, that's an if, and they have kids of their own, I don't want my grandkids to have to go through transplant or go through this vicious cycle that continues to just attack our families generation after generation. So what I request is the ultimate cure, honestly. I want, if possible, I have a dream that one day there is a cure, and it's through targeting genetic therapies where we don't have to deal with the treating the symptoms anymore. We really can just treat the root cause of all this and allow our community, our families, our kids, grandkids to live normal lives. We're not asking for anything except to lead normal lives. That's my request, and that's why I'm here today. And I know myself, my daughters, my family, we are ready for whatever we can do to contribute. So we raise our hands always to do clinical studies or be included in research. We're here and do our part as much as we can. As at the end of the day, we can all find a cure and have a better future for generations to come.
Vita, thank you for those
so
comments.
much.
We're really happy to have you with us today. I think we have a comment coming up shortly. Are we ready with a comment?
We are.
Okay, we're going to hear a comment and then we're going to come back to the panel in just a second.
So we have a patient who called in after having a septal myectomy. They say, I have had a septal myectomy and my HCM is stable. Before my surgery, it was almost impossible to live my daily life. I couldn't lift more than a gallon of milk without being short of breath. I was always exhausted. I had to be put on Norpace because my heart was beating hard and I could hear my heartbeat. I was very aware of my heartbeat. It was scary. My anxiety was horrible. I found a center of excellence and had my myectomy 4 years ago. Everything is still good with my heart. I have a new lease on life.
Well, that is a very effective therapy for some people. Unfortunately, for some, and some of the hearts that I had in the collection earlier have had myectomies and gone over to transplant later in life. So we're glad that you're feeling well now, and we hope that stays for a long, long time. But we need more because we're only taking out the muscle. We're not fixing the muscle itself. So going back to our panel, I want to jump down to Alex. We haven't heard from you yet. If we could have a little understanding of your disease process and what you've done so far and what your hopes for the future might be.
Hi, yeah, thank you so much for having me. I'm Alex. I'm still reeling from Fida's story. I mean, that was just so incredibly poignant. So I'm 32 now. I was diagnosed 8 years ago at 24 with arrhythmogenic cardiomyopathy. And to talk about what I hope for the future, what I've seen in my time so far is I've had pretty much every medication you can take. I have taken or am taking. I've had 4 ablations. I've been shocked twice, then 6 times, then 25 times with my defibrillator. And then I got a different type of ICD. So I feel like I've really run the gamut of existing treatments. So I think what's really important to me here today is to look to the future where there are actual treatments out there, because I feel that as others have said before, these options, while I'm very grateful for them for keeping me alive, and keeping others alive, I don't feel that they're really treating my disease. They're treating my symptoms. They're allowing me to live through these traumatic moments of sudden cardiac arrest, but I'm not really in a position to thrive. So for my hope from this panel, from the FDA, from those that are listening, is to find and continue to explore treatments that will allow not just people with ACM, but all of the cardiomyopathies to really kind of move to I don't know if I can say, but move on from this chapter in our lives to find some sort of, if not cure, but steady treatment that is more consistent than just medications and defibrillator shocks when the medications fail and all we have left is a shock or 25 to, keep us alive. So thank you again.
Thank you, Alex. We've dropped a word in this session that I don't normally use, and that's the C word. The C word, cure, is a tough one, and for our folks over at the FDA and our pharma folks and our scientists who are watching, we understand we cannot cure completely, but we can treat the underlying genetics. So somebody taught me a term a couple weeks ago, and I like it, and I think it works for our community. We want our cardiomyopathies to be undetectable. We want to diagnose it, we want to see that it's there, and then we want a targeted treatment that we know that it's there and we have to be managed and treated for it. But maybe if we have RNA-based or DNA-based therapies, we can get the muscle to work more normally. And that's really our goal. We just want a normal functioning heart. And we're willing to take the steps to do that. And to us, that would be as close to a cure as possible. And that I think everybody I've been talking to over the past couple of months preparing for this, we have a hard time with the C word. So we want to be aware that we can't have that as our complete goal, but to be undistinguishable, undetectable for our cardiomyopathies would be a really great place to see our therapies. We do have a phone call coming in from Jane. I hope we can answer some of her or hear some of her thoughts, but I don't know that we're going to be able to answer her question about off-label use of a drug. So Jane from Missouri, please jump into my ear. Hello, Jane? Is Jane here? I don't hear Jane.
Yeah, can you hear me now?
Now I can hear you, Jane. Thank you so much. What's your comment?
Yeah, sorry, my cell service was cutting out. Yeah, my heart goes out to everybody struggling with this disease, the parents with the children, and it really makes me sad. But I'm 67 years old. Over 10 years ago, I started having symptoms. I have dilated cardiomyopathy, and a year and a half ago, I found out I have the desmoplakin mutation, like Christy, who also shared. And I guess I can't talk too much about off-label use of drugs, but I'm a retired research specialist. I've read a lot, and I'm just hoping that the people out there that are doing the research trials, the clinical trials, can pick up on basic science, cellular research that's out there that may help with my condition and other cardiomyopathies out there. So that's all I wanted to say about that. But as far as my personal journey, if something causes angina for me, heart pain, then I know to avoid it. So over these 10 or more years, I've stopped caffeine, I stopped any alcohol, CBD, even AIDS. All these things have given me heart pain. And there's certain herbs that have given me heart pain. And so I think what I'm trying to say is my own body is telling, when my tells me something, I can listen and do something about it. And I'm taking a lot of supplements. I'm taking resveratrol for my heart, coenzyme Q10, nicotinamide riboside, which is an energy source for the heart, for NAD, and fish oil. Probably a few others, but basically, I'm trying to do everything that I can to try to improve my situation. And I'm just lucky that I have been sort of stable. Unfortunately, 4 times I've had COVID now, and the recent time was just a few weeks ago. So COVID has had a bad impact on my heart, unfortunately. It doesn't put me in the transplant list, which is good, but my heart's gotten weaker. Every time I've had increased PVCs and increased ventricular tachycardia after COVID, increased heart pain, angina after COVID. So I know it's one of those things that's out there in the world, but it's tough that we have to deal with a genetic mutation and other bad things in the world like COVID. And I think that's all I wanted to say. Just feel for everybody out there who's dealing with this.
Thank you, Jane, for your comments. And we heard earlier today also that even getting the flu when you have cardiomyopathy can actually be deadly. So COVID, flu, whatever the virus is, we are impacted more harshly. And sometimes it can be quite unpredictable as to who's going to have a really bad outcome from that. We're going to move over now to Shannon. And I'd like to hear a little bit about your story. We haven't heard from you yet today. I'm holding Julia.
Hi, yeah. Yep. Hi, I'm Shannon. I live near Seattle, Washington. So my cardiac symptoms started like out of the blue. When I was pregnant with my second daughter, I had racing heartbeat almost to the point of passing out. After visiting the ER, I learned I'd been experiencing VT episode, which looking back is pretty terrifying. I was hospitalized and diagnosed with HCM, and an ICD was implanted immediately after I gave birth to her. So 5 years later, my condition was fairly stable, and my daughter's pediatrician suggested testing itself and my two daughters to see if my condition was genetic or possibly something that would be passed down to them. So that's when we found out that myself and my youngest daughter, Kyla, have Danon disease. So over the next 10 years, I slowly declined receiving more interventions and medications, right, and surgeries, leading to heart failure and ultimately transplant. So during this time, I was doing my best to keep our family focus away from me and my worsening condition. But there were so many challenges to that, especially having a husband who is a Navy officer and would be deployed on submarines for months at a time. So today I'm 54 years old. I'm 7 years post-transplant. And my daughter Kyla, she's 23. Her heart has started thickening, and she has started to experience some arrhythmias.
Thank you for sharing. We also heard earlier that women with Danon disease are probably not getting the attention that they need and deserve to get to the root cause of their disease. We've spoken a bit in the global community about the boys with Danon, and it's critically important we provide care for them. But I think we need to look at the entire disease spectrum, as you so eloquently have put. Do we want to go to another comment? Oh, wait, we have a phone call. Sean's been hanging out for a while. Sean wants to tell us what's actually working for him. Can we get Sean on the line?
Hi.
Hey, Sean.
Good to be back. Can you hear me?
Yes, I can. Thank you for calling back.
Okay. Yeah. Okay. I'd like to talk about my disease treatment, how well it's working, and just use that as a launching pad for a couple of comments that I have on the importance of early diagnosis and the importance of approval of therapies for some of these CM conditions today that we heard of that don't have therapies. So I'm currently on vutrisiran. Again, I have hereditary amyloidosis with cardiomyopathy and polyneuropathy. I was diagnosed back in 2019, and I started then on patisiran, which is an RNA inhibitor drug, like we heard of earlier. I was on that for several years, and a couple of years ago, I swapped over to vutrisiran, which is an improved version of patisiran. And I can tell you that since I started treatment in 2019, I have every indication, both from my own well-being and also from follow-up exams and tests that have been done on me at, I'm actually treated at Brigham and Women's Hospital in Boston. Everything points to definitely a halt in the progression of my disease. So that's a big win for me. And I'll tell you why. So I want a real quick early diagnosis. In my case, I wasn't diagnosed. I had symptoms building in for 7 years. So my disease was advanced. Fortunately, we were able to halt the progression, but I would like to see people be able to get diagnosed earlier for all cardiomyopathy disease. I think the way that this can be helped is for expedition of the approval process and regulatory process, and putting a priority on approving new diagnostic devices and test mechanisms. My next comment is on approval of medicinal therapies. In my own example with amyloidosis, we've actually, in the amyloidosis community, have made great strides over the last decade in getting medicines approved. We're now trying to find a go-through trial with a medicine that will hopefully reverse the disease. So we have RNA inhibiting drugs, stabilizer drugs. We also have some patients that have had CRISPR done to them, and there's indicators that that's been successful. So I'd like to see that success spread to all of the cardiomyopathy diseases. And it needs to be done for all the people that I've listened to today. People that have conditions that there are no treatments for right now. And most importantly, the stories I heard with people that have small children. That's tragic to see small children have to go through this process when, with enough effort, we should be able to find diagnostic tools and treatments to treat these people earlier. That's all I have.
Sean, thank you so much for those eloquent comments. And I'm going to back up on one of the things that you've just said. There is a population that you haven't seen here today because it's a hard one to find. And these are infants born with either single or double mutations, specifically for HCM and DCM. These children don't live long lives if we can't get them therapies earlier. So there are some patients who really do need gene therapy in those first early months of life to give them an opportunity to grow and to prosper and to live the life that they want to live. So I thank you for giving me the opportunity to bring the babies to the table. The babies need us, and we don't have time to wait for some of the other therapies to work for them. We need stuff for the babies. So that's hard. We got to figure it out in adults first, so it's safe for kids, but we got to get there pretty quickly. Yeah. Thank you, Sean, for calling. Appreciate you.
Okay. Thanks.
We're going to go over to Genevieve, who has a wonderful comment that she'd like to read now.
One of our community members says, our greatest hope for future therapies is treatment that not only manages symptoms but slows or halts disease progression entirely. We hope for advances that reduce fatigue, chest discomfort, and the risk of heart failure, so Ashlyn can build a full, active life without constant fear of transplant or advanced disease. More than anything, we want her future defined by her dreams, not by HCM.
That's beautiful. We share that parent sentiment completely. We're going to jump to Becca, then to Julia. Becca, we've heard from you in your video, and we've met your beautiful children, all three of them. But girlfriend, what do you want for the future?
Hi, thank you so much. And thank you for the panel for being here. It's so important for you, or for us, that you're here for us. And we're really very grateful. For the future, I just want my kids to be able to grow up and thrive. Now, because of their Friedreich's ataxia, that looks a little bit different, obviously. Our first hope, we know that our genetic disease and their age changes things a little bit, but our first hope is that we can find some therapies that's the disease. That's our first hope. If we can just stop the progression of where it's at, that would be great. Reversing any damage would obviously be coming next, and then cure would be fantastic, obviously. But just being able to stop that, and also just having targeted therapies for us and our kids, because they are young, I hope the future allows them to be in trials and to be allowed to take some medicine. So there's trials that are upcoming that are only available for 18 or 16. And for our family, that might not ever be able to work out. So just being able to, as a community, as individuals, as a family, decide what is the right time to be in a trial, I think is extremely important. If we're limited by the age to be in the trial, we might lose a lot of kids. Or by the time that the trial is ready, their heart might be already too damaged to even participate in the trial. Sorry. Another hope that we have for the future is just for Friedreich's ataxia specifically. It's very hard to just speak about the heart because that is not the only area that's being affected. As you heard from Gretchen, there is a neurological and neuromuscular component to the disease as well. So it affects everything from every molecule in your body. People with Friedreich's ataxia can have vision issues, speech issues, loss of hearing, loss of vision, inability to use their arms, their hands, loss of being able to walk, not being able to take care of themselves at all. So time is really of the essence. And if our hope for the future is that we're never stuck in a position where we finally are able to get in a trial or have a cure for a heart complications, but then are faced with the issue of the neurological component at the same time, and never having to make the decision of, is saving her heart going to just prolong her life, but keep her stuck in a body that is truly falling apart and not giving her any good quality of life. So our hope for the future is that both of those things can come at the same time, and that no parent ever has to, or person has to make the decision of saving my heart or saving my body. And if I do one, should I do the other, or just not anything at all? So that's really my hope for the future.
I know when we spoke prior to the event, you shared those thoughts with me. I'm so glad that you have articulated them here to the FDA directly, because it is a challenging thought. Heart, body, if you have a body that's not functioning, is that the quality of life that you hope and want for your child or an individual wants for themselves? So I think these are very real issues that we want to have conversations with FDA about. We want to talk about the gives and the takes, and we want the chance to try and to have a good conversation about when it's right for an individual person. We're going to hear a comment, and then we're going to hear from Julia.
So Juanita from Westfield, Massachusetts says, I am 64 in 2 days. I was diagnosed with HCM MYH7 at age 40, but in retrospect, I was symptomatic as a child and misdiagnosed with asthma. I am on 7 prescription medications and ineligible for ablation. I have followed Dr. Seidman's research closely and pray every day that genetic therapy will help my son, niece, nephew, grandchild, and great-nieces. I would be first in line to be a study participant.
Juanita, we thank you very much for not only your comments, but your bravery and willingness to step up for a clinical trial, which is the absolute perfect segue for our next speaker. Julia, you have a very interesting story. Could you tell us a little bit about your family and then what you've done in the past few years to treat your HCM?
Sure. So thank you again for this opportunity. And it's so nice, but unfortunate in the way that we're connecting with all these beautiful women who share similar stories. But thank you again for the opportunity. My name's Julia. I'm from Canada. I, just like the other ladies on the panel, have a long history of people who have suffered and ultimately passed away from HCM. My mom passed away on the transplant list when she was 42. My grandfather, 36. My great-grandfather in his early 30s as well. So it's affected a long line of us. And I actually had a late diagnosis despite my mom having this disease. I didn't know I had the gene. I was diagnosed in my early 20s. And then at 24, I underwent septal myectomy with an ICD emplacement. Before that, I was having symptoms of chest pain. I couldn't walk more than like 30 feet on a flat surface. I was fainting. I was short of breath all the time. When I ate food, I would get lightheaded because the blood would go to my digestive system and everything, so many bad things. The myectomy did help, but I realized it wasn't a cure. And seeing my mom go through what she went through with her disease process. She had the same thing, myectomy, ICD, eventually on the transplant list, and ultimately didn't get one in time. I looked to my my local cardiologist actually, and I said, "Is there anything that do I have any hope? Like, I want children, but I don't know if I want children if the same fate is going to happen to me as my family." And so he said, "Oh, there's this something called genetic therapy coming down the line." And so he was like, "Don't lose out hope." So I went online at home and Googled clinical trials, and I found one in the United States at the Cleveland Clinic in Ohio. And I signed up to be the first human patient to receive the genetic therapy for hypertrophic cardiomyopathy, the MYBPC3 gene specific. And I did that obviously because I wanted a chance at a different outcome for me, but also for the thought of if I do have kids, they have a 50-50% chance that they would get it. Is that something that they would have to deal with in the future? And then obviously my community, there's so many people affected with HCM, and although this may not be the cure, this particular gene therapy, or maybe it will lead to the cure, or maybe not cure, just like a lot of other people said. It doesn't even have to be reversing, even getting an early diagnosis, especially in children, and stopping or halting the disease process early enough before they develop the thickened muscle, because this is something that happens over time. That would be incredible. And like I said, if I had to do it all over again, I would absolutely do the gene therapy all over again. And I really hope that other people sign up, and that progress moves forward. And I hope that there's other vectors that are available. So if the therapy that I got didn't work, I would have an immune response, but possibly, so I couldn't receive the same drug again, or vector again. So developing new vectors, or maybe the mRNA route would be great.
Julia, I still am in awe. I've known your story for a while now. We've met in person, we've podcasted, but the courage it took for a young lady like yourself to go ahead and say, I'll be first. We've got nothing but admiration and love for you, and we really respect your willingness to put yourself out there. Given your family history, it's clear why you did it, and we're super happy that you're sharing your experience with the FDA today. And we're going to talk a little bit more about hopes for the future in just a moment, but we have another comment to hear from.
We do. It's from the Danon community. She says, "I am a female identical twin that was diagnosed with Danon disease in January 2026. I am eager to participate in a study with gene therapy. My entire life got flipped upside down. I feel like I have zero control over my health and can no longer hope to have biological children or a normal future. This has been a devastating diagnosis, and my only hope is stemming from a gene therapy to slow or stop the progression of this fatal disease."
Wow. Yet again, we're hearing from females with Danon, and I gotta say, they were a very silent minority for a long time, and I'm glad that they're speaking up and hopefully being seen. Hey, research community, hey, biotech, they're calling you. They need you. So we hope that you're hearing the call. Just a reminder, the phone lines are still open for about another 20 minutes at 703-844-3231, and you can call in and we can talk to you live right here. Okay, ladies, here we are. We are at the moment where we're telling the FDA exactly what it is that we're willing to do for clinical trials. So are there things that you would like to see included in a clinical trial that might help inform the FDA on how they might approve a study design in the future? Does anybody want to start, or shall I just do round robin and call on you? Becca, you're up.
Hi. So my kids have been in a few clinical studies, and each have been in 2 clinical trials. So our family is not shy about clinical trials. We do, of course, look through all of the safety profiles before we make any decisions, but also not being in a clinical trial is a risk because their disease is progressive. As far as what we need in a clinical trial, I think one of our biggest wants is that we're looking at many different clinical endpoints and biomarkers. I know that specifically for hypertrophic cardiomyopathy, whenever we get any echoes or EKGs, we are looking as a family, we are looking at all of the different markers, the troponin levels, the different wavelengths, the thickness of the hearts. We're looking at everything just as parents to see if the disease is progressing. And I hope that as clinical trials are coming through, we can look at some of those endpoints as well. And our biggest thing is that when a study is coming through and it has a clean or relatively clean safety profile, even moderate or even modest, barely any kind of benefit is still a benefit, especially if we're hitting these young kids at 5. If they have a tiny, modest benefit that maybe it's only 3 months of walking, if or energy production, but that over many, many years could be a significant increase to their life. So just looking at those clinical endpoints and knowing that a modest or a tiny benefit is a huge benefit to us in the community.
Thank you for saying that. Again, you bring me back to a point. Alex, I'll come to you in a second. So you bring up an excellent point. And FDA, this one's for you. In the hypertrophic cardiomyopathy community, we know that New York Heart Association class is one of the most common endpoints that you utilize. However, when the HCMA asks patients, what is your New York Heart Association class? We ask it in a different way. What is your average day? What is your good day? And what is your bad day? And the reality is HCM patients do not stay in one class. We bounce, as do many other cardiomyopathies. You'll have good days for a period of time, then you'll have bad days, and then you'll have average days. So you're on this bounce. Only 22% are static in one class, and those are either 1s typically or 3s. So they're asymptomatic most of the time, or they're highly symptomatic. They're actually the happiest people because they know what to expect day after day. So if you have stability of class, that is a win for us. That is not, oh, the trial didn't move them up a class, but it kept them in the class. So we all need to come up with a better way to measure how symptomatic somebody is for what period of time, because stability is a win. It is critically important that our friends at the FDA understand that. Alex, off to you.
Yeah, I just wanted to kind of echo what Julia and Becca were saying. Like I'm very much at a point where the only thing left is heart transplant, which isn't really a tenable solution, but I'm at a point where I'm not ill enough for heart transplant. So it feels like a slow march to that. And at the rate that my heart has degraded over the past 8 years, I don't see how I will make it into any sort of, I don't see how I'll have any sort of longevity without a heart transplant. So for me, you know, what Becca said about having just a moderate sort of improvement. I know that I believe there's one ACM treatment out there with a couple different companies, a gene therapy. I actually enrolled to be in the trial, but then I was disqualified because I had a stroke. And for me, that, I guess, short of dying or a stroke, I would be willing to have a risk for a moderate benefit, because if my heart could be, I don't know, let's say frozen in time the way it is right now, even if that benefit lasts for a year, 2 years, 3 years, 5 years, it doesn't need to last the rest of my life. If that's a portion of time that my heart does not get worse, that I'd have fewer arrhythmias that could turn into, you know, at this point, not life-threatening because I have a device, but that won't give me more shocks, won't give me more trauma, won't cause me to have to take, you know, a bevy of more medication, that would be a huge win for me. And I would get to sleep easier knowing that, okay, the adjustments that I've made in my life, I've made my peace with. You know, I can live with them and they're not going to get worse for a period of time. And that might put off the heart transplant long enough that I might be able to live a normal life, well, within the confines of my disease, a normal life, you know, till without a heart transplant, because I think, you know, that's not really a solution. That's the end. That's live or die at that stage. Like we've said, it's not a guarantee. And unfortunately, we've had instances of people in our communities that have passed away in that process.
Many die waiting, many die of complications early, and some die of complications late. Transplant is fantastic. I'm 9 years out and I'm doing great. However, as I sat here waiting for this meeting to start, I got a call from my team. They said, we don't like that number. We want you to come back in and do more testing. So we're always on our toes. We're always waiting for the next shoe to drop because you don't know what's coming next. So transplants are great, and they are only available thanks to clinical trials and the ability of our global community to say it's worth the risk. So let's take it another step. Let's say the genetic therapies, the RNA-based therapies are worth the risk. So if it's worth the risk to the individual family, great. If it's not, step away. When we've done focus groups asking HCM patients, are you interested in genetic therapy, or do you think it's a viable option? Every single one says we need to research it. We need to find a way. Would you step up for a clinical trial? A lot of people said I would do it, but after others went first, like brave people like Julia. So we know that they're interested. We know that it is a destination we hope to arrive at someday, and it's going to take us all to get there. We have a comment coming up. Off to Genevieve.
Yes. Thank you, Lisa. We have so many excellent comments talking about, the need for better therapies. One of these is talking about cardiac fibrosis, and the commenter says, my primary goal and greatest hope is to find a treatment for cardiac fibrosis, specifically something that can treat and reverse existing fibrosis. I urgently need effective treatment options that do not just manage symptoms, but actually treat the fibrosis itself.
I couldn't agree more. You saw pictures of my heart. There's a lot of fibrosis and scar in there. And we now know that we can quantify scar through MRI. We're doing a great job at that with all our cardiomyopathies. But how do we stop it and how do we reverse it? And I'm happy to say there are some trials on the horizon that are pinpointed at that exact destination. So we have the scientific community behind us here. Now we just have to see if it works. And we start with the phase 1 and we go from there. So stay tuned and we'll give you some more information on that. Does anybody else? Oh, Shannon, please speak up.
Yeah, so being part of the Danon disease, family, I've shared all my medical records for natural history studies. So because in Danon disease, it varies so much between males and females, but also within female community, at what time in your life you're affected and when you might get to the point, like Alex was saying, that you need a heart transplant. So some people in their 20s, I was in my 40s. So it varies a lot. So those natural history studies are very important. Kyla, my daughter, she's currently participating in an observational study because she wants to help advance knowledge about how Danon progresses. She values the connection with her study team and sees this as a step toward potentially participating in future clinical trials. We know that there are clinical trials in males right now. And so she's hoping for a phase 2 gene therapy study in females as soon as possible. It's really amazing because she holds hope. I mean, a lot of stories, there's fear and stuff, but she is really clinging to the hope of being in a trial. She's like, she knows I'll be in a trial and, things will be better. So that's amazing. If she doesn't need to go through what I did, that is my big hope for the future.
I think we all share that for our children, grandchildren, or whatevers they are, the next generation. Does anybody else have a comment that they would like to share about their hopes for gene therapy or RNA-based therapies in the future? I'm going to go to a comment while you guys think.
So our next comment is, as a young heart transplant, there are worries about getting cancers and infections from the long-term use of immunosuppressive medications like tacrolimus. In order to live to an old age, I would need to have several heart transplants. Given the limited supply of hearts, will that be an option when the time comes? I don't like the many restrictions that come with now being immunosuppressed and knowing my longevity will be compromised.
Wow, that is a tough issue to face. We do get these new hearts. We go on things like Tacro and other immunosuppressant drugs. We can live well with it. But there's always a risk, and you never know what's around the corner. So we would love better therapies so we can keep our hearts longer, and as I call it, go the whole ride with one heart. Julia, please.
Hi. So I guess one thing that I would like to see with future gene therapies does have to do with gene therapies, but also the research and development of different immune suppressant medications for this reason during the gene therapy process with the vector anyways, that you do have immune suppression for a period of time. And I just feel like some different, immune suppressing drugs would make the process safer and more streamlined. And I think more people would sign or be more encouraged to participate if some of the risks associated with the unintended effects were lessened. I personally, I feel well right now. And like I said, I would do it again. And I was excited to participate. So excited that I researched and found it myself. And I think that there's many other people who are in a position like I am, where I don't have children yet, or even if you do, that they would sign up for something to potentially help themselves, but ultimately progress science and hopefully make people's lives better. But then also, I would also like to see different vectors, like I said, be used, more vectors, so that people who have already had a gene therapy in a clinical trial could possibly have a second round, because I would do a second round if given the chance. And also the mRNA for people who maybe don't want something as permanent as a gene therapy. There's something new coming out where it would be like a bi-monthly injection or something like that. I'm not really sure how it works, but it would make your cells potentially create the protein that it needed for a period of time. I think that is super promising. So I would love to see lots more in those areas.
Thank you so much. And I'm going to go back to Becca for a minute, because we had a conversation before the meeting about the complications of certain vectors being used, and a very interesting issue if you have multiple family members with a genetic mutation in the family, all of which that might need gene therapy at some point, but they might become in contact with the vector or the virus and become immune. So you talked very eloquently about that. You want to share your thoughts?
Yeah, so from my understanding, and I am not in research at all, just a dedicated mother who's done research, but we found out that there are some therapies upcoming or potentially down the road. If one person in the family gets a vector, they have to not see the other person who might need the vector, because of risk of the second person then getting antibodies for the vector. So for our family in particular, how would we decide which kid gets it and which kid doesn't? Or if for us, it would be very important for drug companies coming up to definitely have that in their mind, for families who have many people in the family with that disease, to either lower age ranges or make it available for both family members to be in the trial at the exact same time. That way families don't have to be split up for an extended period of time. That would be extremely important for our family, to make sure that we're all together and not having those complications, deciding who gets what when.
Thank you so much for that. Do we want to go to another comment?
Yes. Our next comment says the greatest hopes would be for 1, therapies that will reduce the risk of sudden cardiac arrest; 2, therapies that will reduce symptom burden, especially exercise tolerance and ability to participate in sports; 3, therapies that are safe in pregnancy; and 4, therapies that could prevent the development of HCM in any gene-positive children.
Those are all very good aspirations, and we share them. Who else has a comment? I know I got a group of women here. They're all going to have opinions. I'm very sure of that. Alex, let's hear what you have to say.
Yeah, just kind of going off of that, I think something that we haven't maybe touched on as much is aside just from therapies. Someone mentioned this before, but a lot of the medications that we take are cocktails that are adapted from more traditional types of heart failure or more traditional coronary heart diseases. So if there were in fact more targeted medications for our specific needs. I think that is not such a step as, gene therapy, but that would be great. Like I take a whole bunch of heart failure medicine, but they're only for left-sided heart failure, and they don't really necessarily know how they will work on right-sided heart failure, but I have right-sided heart failure. So that's just an example of that. I think that in addition to, obviously gene therapy is much more exciting and maybe sexier than to talk about than medications, but I don't think we should forget about the fact that we are taking medications that are, I don't want to say unproven with our diseases, but we were not the intended users. So and it's a little bit of like a square peg in a round hole sometimes, or you're kind of blanketing over the disease and getting all these side effects that maybe a more targeted medication could eliminate some of these problems that it seems like really all the cardiomyopathy patients face, not just one particular disease.
I think it's a great point. There's some new data coming out in the HCM community related to beta blockers. And guess what? They really don't work as well as we thought we as the patients knew they weren't really working, and they were just making us pretty miserable. But now the science is proving that, because we have something to compare it against in HCM, and that's the myosin inhibitors. And when the Voice of the Patient report comes out, you're going to see what the burden of disease still is to the beta blocker community versus the myosin community. It is literal night and day. And as this was a genetically based PFDD, I did not include the information in our survey results today, but it will be in the report. And it is fascinating. Similar results come into the amyloid patients who are on targeted therapy as well. And unfortunately, that has not reached the rest of the cardiomyopathy community yet. But we are getting there, and I'm hoping that as HCM is the largest population here today, we're going to learn and we're going to hand it down to our little brothers and sisters all over the cardiomyopathies and help everybody get to the therapies that they need. We're going to go to one more comment.
Yes.
And we've got 3 more minutes, so last comment's coming up, ladies.
And building off of the beta blocker comment, we had one come in that says how the medications, especially the beta blockers, have lowered my quality of life. I now live with extremely low blood pressure, on average 85/56. At what point does a patient's quality of life become more important than a beating heart? I feel like that has been lost with this disease.
I couldn't agree more. And this is the burden of disease, and these are our hopes for the future. Having experience going into heart transplant myself, you had to be sick enough but well enough, and it's this window, and it's not a guarantee. I had blood pressures in that space when I was heading into transplant because I was actively dying, and we stopped that with a transplant. Thank you for the technology and all the clinical trials that made that happen, but there's too many people suffering for too long, and there's simply not enough hearts, and it's not the solution. So proof yet again that this is what we need. Final comments from Fida, and then from anybody else. We got 2 minutes left. What do you want, my love?
I really want to echo everybody's thoughts and hopes, and requests. And I want to, the use of the medication, as we talked about, did they improve the pills and the metoprolol and all that? Did it improve quality of life? No, really, they didn't. They managed the symptoms, maybe a little bit. All I know is my daughter was sleeping 12 hours out of the 24 hours instead of playing outside, going on a bicycle, or even being able to go to school. I just feel like the what's available right now, I don't know how truly it is improving our quality of life and how truly it is improving the symptoms. Except for the ICD that shocks you back to life, I don't think whatever we have is really improving our quality of life or giving us longevity or anything like that. We just sit there and watch our life slip away, sleeping, tired, no energy. We're not productive in this society. We watch everyone being productive, and we want to be productive. We want to contribute, and feel useful, and we just can't. So back to back to where, the beginning. We just need better than this. We need more targeted genetic therapy. We need something much more efficient than what we currently have.
Vida, thank you. And we're going to give our last comment to our brave pioneer, Julia. You're on mute, hon. You're on mute.
Thank you again. And I'd just like to say, I think, to summarize what I heard from all the women today, and the reason why I went into the gene therapy trial is because even if I didn't come out making me better or working, it gave me hope. And now I've done it, and I don't know what my future holds now, for good or for better or for worse, but that is hope that I didn't have before. Before, I felt like I was on a set path to be exactly like my mom, my granddad, my grandfather. But now with this, I'm being followed for 5 years, and we have been given hope. And I think that that is such a big thing that you can give us by approving these therapies and by expanding clinical trials.
I think leaving on the title of hope is a great way to end this segment. Ladies, thank you all for your contributions. We truly appreciate each and every one of you. We are going to then pivot back to Joey to review some of our responses from our online survey. And Joey, off to you.
Yeah, thank you, Lisa. And really what we're doing here is just pulling out and talking about all of the information that we gathered from this survey. The respondents shared a mix of concerns and hopes regarding living with or being at risk for genetic cardiac disease. Many highlighted the fear of the disease progression and sudden cardiac events, the daily living limitations. And we heard a lot about the emotional toll, including anxiety and depression. Hopes for the future centered on gene therapies, curative treatments, and improved quality of life for themselves and their families. Some expressed worries about passing the disease to relatives, challenges with healthcare access, and treatment affordability. So overall, the responses reflected a desire for therapies that address both the physical and psychological burdens, with a strong hope for transformative advances. So we'll leave you with this word cloud, all right? A 10,000 look down at what the takeaway words are. Just take a look at the words on the screen here. We see concerned, symptoms, fear, children, worry, HCMA, family, disease, but we see medication. And I want to leave you with this: future. Everything lies in the future. We move forward from here. We thank our participants for participating in this survey and dedicating their time and energy so we could have this conversation today. So a nod to them. Lisa?
Joey, thank you so much for sharing your big heart with us today and giving us those survey results. And here we are with the close. Here are my final comments. We want to thank all of you for joining us today and learning more about genetic cardiomyopathies and gaining a better understanding of our hopes for the future to keep our families safe from the consequences of these devastating diseases. Today would have been my father's birthday. He would have been 91 years old. He passed away in 2008 from complications of HCM. My father performed CPR on his father in 1953 when he had a sudden cardiac arrest from then undiagnosed HCM. He didn't survive. He then went on to lose his brother at the age of 47 in 1990. And 5 years from then, he would do CPR on his daughter, my sister Lori, who had a cardiac arrest at the age of 36. Sadly, she did not survive. And she became the inspiration for what you now know to be the Hypertrophic Cardiomyopathy Association. You have heard today from countless families dealing with similar grief, picking up the pieces after far too many losses or traumatic medical experiences. However, our voice is steady and clear. We need precise medicines and therapies that hit the root cause of our disease processes. Our hopes are not simply aspirational. We are willing to do the work side by side with researchers, pharmaceutical companies, regulators, government officials, and whomever needs to better understand the problem and be part of the solution. We, the patient community, will participate in well-designed clinical trials. We will show up. We look forward to a day to work with every member of our complex ecosystem to bring about meaningful change to families with genetic cardiomyopathies throughout the world. I want to thank our partners at the Amyloidosis Research Consortium, the Friedreich's Ataxia Research Alliance, the Dilated Cardiomyopathy Foundation, the SADS Foundation, and of course my team at the HCMA for all of their very hard work and dedication to bring this program to you today, and the work that we will be doing in the months ahead to create the Voice of the Patient report, to put all these amazing contributions and our survey results down on paper so that you can all use them to build a better future for all of us. I want to give a big thanks to Dudley Digital for their production work and planning and all the behind the scenes today that made us look very professional sitting up here. And once again, a big thank you to our medical advisors, Professor Watkins and Dr. Seidman, for their oversight during this process. And finally, a huge thanks goes to our entire community for bringing together a united voice. Thank you for bravely sharing your lived experience and what it is like to survive with genetic heart disease. Our big, strong, or weak hearts, all thank you. This meeting has been so impactful, and we couldn't have done it without each and every one of you. In the coming weeks, we'll compile the information from today, and the survey will stay open for another 30 days, including the polling data and the comments, all into our Voice of the Patient report, which we hope to become available estimated October 2026, and will be available on all of our partners' web pages. If you want to still submit written comment, you can do so today. If you're watching this after live production, you can contact us through our office. Today's program will be available on demand immediately following this meeting, and you can use all the same links. Lastly, it's been my honor to support this meeting, which I have hoped that will have a lasting impact on the future of genetic cardiomyopathy treatments and development. So once again, to the entire community, thank you for making your voices heard today, and thank you to my co-hosts, Genevieve and Joey, for their wonderful help and making this fun and entertaining to do, even though we're talking about a hard topic. And we are done. Thank you so much.